Okay.
A very good morning to all. Welcome to the sixth Accelerate Weekend. Today,
Also, he has several awards. This introduction, I would like to invite Dr. Sunil Kumar giving his wonderful talk on variable high pressure
NMR spectroscopy attachments of NMR spectroscopy. So, how to determine the structure of small molecules, peptides, proteins, and all these kind of aspects. So, today we'll discuss about going into the details. So, before going into the details of the talk, I would like to introduce my institute. So, this Institute for Integrated Programs and Research in Basic Sciences, Mahatma University, in 2009, and we offer integrated master courses in 5 different subjects: chemistry, physics, biology, computer science, environmental science, and doctoral degree also. And we have a very good instrumentation facility, including a 400 megahertz, spectrometer, HPLC, UVIR, AAS, GCMS, this kind of instruments are there. So in addition, I would like to convey another message. So in the year 2020, a group of people, we constituted a society called Magnetic Resonance Society Kerala. This is to bring together all the persons who are interested in this field. So, if we have already more than 150 members in this society, if someone is interested to join, and we are frequently organizing workshops and seminars and all these kind of training programs. If someone is interested to join this group society, you can contact me, Dr. Dinesh. He is the president of this society. Okay, now we are moving to our topic. So, high pressure science. So, often, always we want to run away from pressure. But when pressure acts, when we consider pressure as a thermodynamic variable, so there are many branches of science related to pressure. So we have high pressure NMR spectroscopy, high pressure micromolecular crystallography, high pressure fluorescence spectroscopy, high pressure small angle X-ray scattering, high pressure food sterilization method is also there. And all these branches are well described in a textbook edited by Professor Kazuki Akasaka and Hiroshi in high pressure bioscience, basic concepts, applications, and approaches. A textbook published in 2015 by Splendor. And I was working with this Kazuki Akasaka in Japan. And I started my high pressure NMR studies with him, who only developed these methods. Okay. So now, coming to the topic. So the dynamism of life, it originates from the dynamics of molecules, particularly proteins. So the function of a protein, it depends, or it is very closely related to its dynamics. And a static protein will not be functional. A dynamic model of the protein is necessary to explain the function of it. For any molecule it is applicable. So here in this video, you can see that this is the structure. This movie is showing the catalytic function of dihydrofolate reductase. It's an enzyme which catalyzes the reduction of DHF, the dihydrofolate, into tetrahydrofolate. See, during its function, the protein is moving. So without this movement or without this mobility or dynamic nature, it cannot be functional. So this variable high pressure NMR, it's a very advanced technique for studying the protein conformational fluctuations in a wide time scale, starting from 5 per second to a second. So this is nothing special. So we are applying pressure to the protein system or protein solution. And then we are observing the changes happening to the protein conformations by the application of pressure using high resolution NMR. And we need a special experimental setup for this. And we will come to the details of this experimental setup soon. So we can use our normal spectrometer for high pressure. But the sample tube and other setup we need, especially different from our normal NMR experiments. So coming to proteins. So proteins in solution, they exist as a mixture of different conformation states, and they are in dynamic equilibrium. They are exchanging between each other. So when we do normal conventional NMR spectroscopy for determining the structure of a protein, we determine the population weighted average structure. Population weighted average. So we will get a structure which is very close to the mass populated conformation of the protein, which is the basic folded state. And we don't get an information about the low populated conformations. So in the protein solution, there will be a basic folded state, which has the highest population, maybe 80%, or more than that. 90% depends upon the protein which we study. And there will be other conformation states with a low population. So using normal techniques, we cannot detect these low populated states. We call them low populated hidden conformation states. So similarly using X-ray crystallography, so we are crystallizing the most populated conformation, the basic folded state of this protein. And then we are determining the structure. Here also we are neglecting these low populated conformation states. And this most populated conformation may not necessarily be the functional one. The low populated may be slightly disordered. They may be the functional one. So it is very important to detect them and characterize them. So using this high pressure NMR, we can populate and we can increase the population of this co-interruptions. Correct. Okay, so we need to understand the identify and characterize these low populated conformation states in a protein which may be functional ones, so functionally important. So this high pressure NMR is a very good tool for the identification and characterization. So before going to the examples, so I would like to mention about what is the effect of pressure on protein conformation. So when we apply pressure to a protein solution, mainly two things are happening. First, so as I mentioned, there are more than one conformations in a protein solution. So the first one thing which is happening by pressure is the general compression of a single conformation. So as I shown here, we have two conformations. The same conformation is undergoing some kind of compression. So another important thing what is happening is the equilibrium shift from one conformation state to another conformation state by the application of pressure. So say in the left part of the figure B, the round-shaped conformations are in a larger population, and the other one, the yellow-colored one, which is the low population. But we applied pressure to this system, and the equilibrium shifted to the right part. Say the yellow-colored ones, the population, the number of conformations, yellow conformations, was increased. So now we can detect them using NMR, and we can characterize them using NMR. So this is what we are doing in the high pressure NMR spectroscopy. So how these changes are happening? When we apply pressure to a high pressure protein solution, as I mentioned, two important things are happening. It's a compression of the same conformation state, and the other one is the shift of equilibrium from one conformation to the other. So these things can be explained based on this volume theorem. So as we all know, when we apply pressure to an equilibrium system towards the lower volume side, it's called inertial limits. Okay. So in a protein conformation equilibrium, different conformation states which are in dynamic equilibrium, they are having different partial molar volume. So when we apply pressure to this protein solution, the conformation shift will happen from a conformation state with a larger volume. Usually this is the basic folded one, and to the lower volume side, the other conformations which are low populated. Okay. And this is how the shift in equilibrium from one conformation state to the other one is happening by the application of pressure. So from a higher volume conformation to a lower volume conformation. So the other thing, the compression of the same conformation state. So this conformation, conformation substate before. This is not a single, it's an ensemble of conformations with a comparable energy and a slightly different partial molar volumes. So when we apply pressure to this single conformation substrate, the transition between them within the same substrate, the substrate will happen from higher volume to lower volume. So this is responsible for the general compression of the same conformation state. So based on this volume theorem, we can explain how what are the things happening in a particular in a protein solution by the application of pressure. So all these changes can be monitored with the help of high resolution NMR spectroscopy. So in this potential energy diagram, see the partial molar volume is largest for the basic folded state, the lowest energy conformations, the most populated conformations. So when the structure becomes disordered, then finally coming to the fully unfolded state, which is coming at the top of this potential energy diagram, it is having the lowest partial molar volume. So volume is increasing when we go from top to bottom, and the energy is increasing from bottom to top. So this is so. So after my stay in Japan, I went to University of Regensburg, Germany, and this is there also we did many high pressure number studies proteins, different proteins. We will see some of the examples. And this is the high pressure NMR setup in Germany. See, so all of you know the normal NMR2, which is made up of quartz or glass. So here we are using a special NMR2, which is made up of ceramic, ceramic NMR2, which can withstand high pressure up to 3,500 bar. So experiments or works are going on to increase the pressure limit. Okay. And we have a pressure generator using which we can generate the pressure. And this pressure can be transmitted to our protein solution taken in the ceramic cell. And the pressure mediator, which we use for transmitting pressure from the pressure generator to the protein solution, is usually we use water. And we separate them, the protein solution and the pressure mediator, using a membrane. Okay. And this ceramic cell can be inserted. So on the say, so this ceramic cell is connected to the end of this pressure line, and this pressure line together with the NMR sample tube is inserted into the magnet. So in this university we have many 5 spectrometers, and this 800 megahertz spectrometer was used for the study of these proteins. And we inserted this high pressure ceramic cell together with the protein sample into this magnet. And there we have 2 types of pressure generators. One is the manual, and we have to rotate some piston and change the pressure from 1 bar to 10,000 bar or something like that in regular intervals, 100 megapascal bar, 200 megapascal bar intervals that we can decide. And there is another automatic pressure generating system. And this is controlled by some computer programs. And we can set up the pressure range and pressure intervals. And it will automatically change, and it will automatically control the pressure. And we can record the NMR spectrum, 1-dimensional, 2-dimensional, higher dimension, at various pressures, automatically using this automatic pressure generator. Okay, this is the experimental setup. Okay, and the spectrometer is a normal NMR spectrum. Okay. So then what are the NMR parameters which we analyze or observe as a function of pressure? Suppose we have an unlabeled protein. Now all of you know what is a labeled protein and unlabeled protein. So the carbon 13 is the NMR active isotope, but the 99% is carbon 12. So when we come to proteins, we have to label with the C13 for increasing the sensitivity. And similarly, we are using N15. We are observing N15. The nitrogen with the high value of 1/2 for studying for studying the nitrogen nucleus. So its population, natural abundance is also very low. So we have to spin label both of them. So suppose we have an unlabeled protein, in the case of small proteins, is required. So we record different one-dimensional proton NMR spectra as a function of pressure, and see different spectra displayed here, arrayed here. And then we can analyze the peak position, proton chemical shift. The peak position is changing. And also another thing, the peak area or peak volume, in this case it is peak area, it's also changing as a function of pressure. And we monitor or we analyze all these kind of changes as a function of pressure. So when we come to a labeled protein, suppose we have an N15 labeled protein. So we can record proton N15 H species. And each spot in this two-dimensional spectrum, one axis proton and other one is N15. Each spot represents a single amino acid, the backbone NH2. So see, this is from ubiquity. So see the spots, the cross peaks are changing, the positions are changing as a function of pressure. And some of them are changing, the volumes, the peak volumes are changing as a function of pressure. So we analyze the peak positions as a function of pressure, from which we can obtain the change in the proton and N15 chemical shifts as a function of pressure. And similarly, we can monitor or we can analyze the peak volume changes as a function of pressure. So these kind of things usually we do. So what are the different types of pressure-induced spectral changes we usually observe? So one, change in peak position, chemical shift, without any area or volume change. Say two different types of position changes in the cross peak corresponding to a particular amino acid. This is a representative, a schematic representation. So we can have a linear change of position, and we can have we often observe a non-linear change in the position of the cross peak. And both of them, they have different meaning. So the linear change in chemical shift. See, the peak volume or peak area is not changing. All are having the same area or volume. And the linear change indicates fluctuations within the same conformation state. As I showed in the previous slide, there are different conformation states in a protein solution. And the same conformation can fluctuate. And this linear change, this chemical shift indicates that the fluctuations within the same conformation state. And the non-linear type of chemical shift change or peak position change, it indicates conformation transition from one conformation to the other one. The equilibrium is shifting from one state to the other one. This is indicated by this non-linear change. Okay. Say another thing is also we have to consider. So this here, the equilibrium is shifted from state 1 to state 2. And this nonlinear type of change is happening because the population of state 1 is decreasing and state 2 is increasing. But we cannot see the chemical shifts from state 1 and state 2 separately because they are in fast exchange. Okay. But the equilibrium or population is shifting. by the application of pressure. So another kind of spectral change which we observe by the application of pressure, the peak volume or peak area is changing, but there is no change in chemical shift. The position is not changing. Say, so here a cross peak from and HS gives you cross peak from a particular amino acid, and the peak volume is decreasing, but the position remains the same. And we can plot this peak volume as a function of pressure, and finally it will go to zero. The second peak, a new peak may appear, and the volume of the new peak will increase alongside with the decrease in the peak volume of the original peak. So the second peak may not be visible because of different reasons. One reason, the second peak may be overlapped with another peak. In that case, we cannot see. And also the second peak may be very broad due to conformational exchange or something like that. So in that case also we cannot observe the second. But we can observe the original one, the change in the peak volume. And this type of spectral change indicates the occurrence of two or more conformations in the equilibrium, which are in slope exchange, and their populations are changed by pressure. Okay. So another kind of spectral change by pressure, which we usually observe by pressure, it is both chemical shift or peak position and peak volume are changing. So here, position is changing linearly. At the same time, the volume also decreasing, and nonlinear change also happening. And this type of spectral change indicates the occurrence of more than two conformations, some of which are in fast exchange. The chemical shift change is indicated by the conformational transitions between different conformations which are in fast exchange. And the volume change indicates the conformation transitions between different conformations which are, and the equilibrium shift is happening due to the application of pressure. And another kind of spectral change which we observe is the peak width. The peak width may increase as a function of pressure, a change in peak. This type of spectral change, change in the rate of exchange between conformations by application of pressure, and the rate of exchange between different conformation states, it contributes to the peak. Okay. Then coming to the fast conformation fluctuations within the same folded state. As I mentioned earlier, so there are different types of chemical shift changes as a function of pressure. One is linear one, and the other one is non-linear. And the chemical shift change, either linear or non-linear, as a function of pressure, represents conformational fluctuations or conformational transitions which are in fast exchange between conformations which are in fast exchange. So this linear chemical shift change, it indicates conformational fluctuations within the same folded state. For example, the basic folded state, which is fluctuating. So with the application of pressure, so linear changes, we can measure this delta delta. This delta delta is determined by the flexibility, how much conformation fluctuation is not, the extent of fluctuation. Okay. And the nonlinear time, as I mentioned earlier, transition from one state to the other, state 1 to the state 2. And these two, state 1 and state 2, are in fast exchange. This is a two-state model. So in some cases, when we move further, we can see that more than two states may occur. in the real system. So the plots will be different. So we will see how it will become. So we can determine different thermodynamic parameters between the state 1 and state 2 using some thermodynamic equations with the change in chemical shift as a function of pressure. So the delta G0, the free energy difference between state 1 and state 2, and the partial molar volume difference between state 1 and state 2. We will see different thermodynamic equations later, which we can fit to these chemical shift changes as a function. Then we can get this delta G0 and delta G0 values. So these are some examples in which there are three states, three conformation states are existing, which are in fast exchange. And state 1 is going to state 2, and state 2 is going to state 3. And here also, it is going from state 1 to state 2 and state 2 to state 3. Okay. And all of them are in fast exchange. So, as I mentioned, so we can have different types of chemical shift changes, linear or non-linear. So the linearity or non-linearity of these chemical shift changes as a function of pressure can be estimated by fitting this equation to a plot of chemical shift changes as a function of pressure. And this equation delta I is equal to AI plus BIP plus CIP squared. Here delta I, here P is the pressure, and delta I is the chemical shift of the ith residue. AI is the chemical shift of the same residue at normal pressure, 1 bar. And BI is the first order coefficient in the pressure dependence. It is expressed in parts per million per bar. And CI is the second order coefficient. It represents the non-linearity of this shift change. BI value represents the linearity. Okay, so we can estimate the linearity and non-linearity of a chemical shift change by fitting this equation into it. We have different programs for fitting this. So conformation fluctuations in slow time scale, greater than millisecond scale. So in that case, we can observe peak volume is changing and the original peak. So by increasing pressure, we can say that the volume of the original peak is decreasing. Suppose we can see the second peak, another peak will also appear corresponding to the second conformer. And the volume of the second conformer will increase as a function of pressure. The first one, the original one, the volume of the original one is decreasing as a function of pressure. And the volume of the second one, corresponding to the second conformer, it will increase with the application. That means the population of the first conformer is decreasing with the pressure, and the population of the second conformer, it is increasing. And when we come to the peak volume, if at the same 200 megapascal or 2000 bar, if we sum up the volume from the two peaks, original and split peak, and we can compare with the other pressure, the total volume will remain the same if there are only two configurations. Okay. So coming to the thermodynamics of variable high pressure NMR. So here we are considering a protein system with two conformations. The native state, we call it as I state. The equilibrium constant between them, they are in equilibrium, thermodynamic equilibrium. So the equilibrium constant K is equal to concentration of I divided by concentration, which can be again equal to e raised to delta G. So delta G is equal to delta G0 plus T delta V0, where delta G0 is the free energy difference between the two states, N and I states, at normal pressure. And delta V0 is the volume difference, the partial molar volume difference. Okay. So we can plot delta G as a function of pressure, the free energy difference at normal pressure. And the slope of this plot, it is in most of the cases, it is usually linear. So the slope of this curve will give the volume difference between the two conformation states. So we have different types of chemical shift changes to which we can apply. Two states, state 1 is going to state 2, and then 3-state models, we already saw all these kind of things. So we have a general equation like this: delta is equal to e raised to minus delta G1 divided by RT. And we can explain it like this for a two-state model, and for a 3-state model, we can expand this to like this, and we can fit these equations to these different types of chemical shift changes as a function of pressure. And we can get delta G0 values and delta V values between different conformation states. Okay, so now when we come to the conformation transitions in slow time scale, so the peak volume is changing. So there are two conditions. So suppose, as I mentioned, only one peak is visible. The other one is not to light broadening or overlapping. So we can plot the peak volume change is decreasing as a function of pressure. And we can fit this equation with the volume at a particular pressure equal to V0 divided by 1 plus e raised to minus delta V0. From this, we can get the delta G0 and delta V0 values between the two. So the second case, both of the peaks are visible, original one and the second one. And the volume of the original one is decreasing and the second one is increasing. So we can estimate the peak volumes. So the volume represents the population. So the equilibrium constant at a particular pressure K is equal to VNP divided by VOP. VNP is the volume of the new peak, and VOP is the volume of the. This ratio will give the equilibrium constant. So we can determine the equilibrium constant at different pressure, from which we can get the delta G0 and the delta V0. So these are the basic concepts in the variable high pressure NMR of proteins. Almost 30 minutes, right? Yeah. So I would like to discuss some examples, case studies. One is CMIP R2 subdomain protein. Other one is RAS protein. Then E. coli, the third one is E. coli DH, dihydrofluoride protein. So, okay. So extensively hydrated, but for a novel state of global. And this is about CMIP, the DNA binding domain of CMIP. And we identified different conformational states, which are very functionally very important in the R2 subdomain of CMIP protein using variable high pressure techniques. And all the results are published in a biophysical journal. And so before going into the details of this uh, research work, so there are two important messages, particularly to the uh, young generation of research group. One message is this: we should not come to a conclusion with only one type of experimental methods applied on the same system may lead to exciting results. So I think you understood. So you suppose we have different experimental techniques which we can apply to the same system. We should do that. We should not conclude with only one type of experiment. You will come to know why I say it like this. And also, suppose we applied two different methods. We may get two different results. But when we combine them, we may get very interesting results. So the folding and unfolding studies of proteins. It's very important. How a protein is folding. The folded state is the functional state from the linear amino acid, the primary structure to tertiary structure. And the reverse unfolding. How this unfolding is happening. What are the intermediates involved? And what are the thermodynamic parameters? And all these kind of studies can be done using different methods. One such method is fluorescence. In the case of proteins, tryptophan fluorescence. So the emission maximum, lambda maximum of tryptophan fluorescence can be plotted as a function of pressure. For example, any perturbation, temperature, pressure, or chemical denaturing. So when the tryptophan, it's a hydrophobic amino acid, and usually it occupies the core region of a folded protein, and it is away from the external polar environment, water flows. When it is in the interior, its emission maximum is around 355 nanometer, 335 nanometer. So when the protein unfolds, all these tryptophans will get exposed to the surrounding water. So the lambda maximum, the emission maximum, will be changed or shifted to 355 nanometer. So we can, when the emission maximum, the fluorescence emission maximum of tryptophan is close to 350 or 355, we can say that they are in polar environment. But the protein is unfolded state. So another method which we can use to monitor the unfolding of proteins is NMR spectroscopy. So we know that there are characteristic peaks, prospects from the folded state of a protein. When we apply some perturbations like pressure, chemical denaturing, or temperature or something, therefore, suppose the protein is undergoing unfolding, a peak volume or peak area corresponding to that corresponding to the folded state, it will decrease and finally go to zero when all the folded states undergoes unfolding. Okay, so NMR is a very good method. Okay, so now we know two important methods for monitoring unfolding of a protein. So in this study, we have applied variable pressure tryptophan fluorescence and proton NMR to safely R2 subdomain at low temperature and to know its conformational changes along the pressure axis. Here the perturbation used for monitoring the conformational changes is pressure. Okay. So CMIM is a transcription factor involved in proliferation and differentiation of hematopoietic cells. And it has three function domains. One is DNA binding, transcriptional activation, negative regulation. And this DNA binding domain, it has three subdomains, R1, R2, R3. And the subject of our present study is the R2 subdomain. It has 52 amino acids. So this is a three-dimensional structure of R2 subdomain. So when we look into it, we can see different interesting things. So there is a large internal cavity, R2 subdomain. And also it is more dynamic, it's fluctuating. And its stability, the thermodynamic stability is less compared to the other subdomains, R1 and R3. Okay. Say, so when we analyze the structure, we can see that this valine 103 residue, it is directly pointing to the cavity. So when we mutate valine 103 with a leucine residue, which is a amino acid residue with a long side chain, we can reduce the size of this cavity. Okay, we'll see what is the effect of this. And see, there are three tryptophans in this R2 subdomain, and all our side chains are covered. You can see that they are in the interior, the core region, the hydrophobic core region, R2 subdomain. So the proteins used in the study, two proteins we used. One is with the cavity. We call it as WT star. There is another reason. There is a cysteine residue at position 130. So this cysteine residue usually makes some problems by the formation of disulfide. Like that. So in order to avoid this, we substitute with the Q130 I. It will not affect its structure. The cavity size is also not. Okay. Then there is the second protein which we used is the cavity filled V103L mutant of CB2 subdomain. And the experimental methods which we used were variable pressure tryptophan fluorescence. The pressure range which we applied is from 3 megapascal to 600 megapascal, or 30 bar to 6,000. And the pH used was 7.5, and the temperature was 5 degrees. Then we applied the variable, then we did variable pressure, one-dimensional proton NMR analysis, and the pressure range used was from 3 megapascal to 300 megapascal. And we used the same pH and temperature, 7.5 pH and temperature of 5 degrees Celsius. And this is the online variable pressure fluorescence system at King University, Japan, where I was in before. So here also we have a pressure generator and we have a pressure line. Here we hold the sample and we can measure the fluorescence at different pressure. And this is the variable high pressure NMR system, the King University. Right? There 600 megahertz glucose. So now coming to the results. So initially we did the tryptophan fluorescence as a function of pressure. From 1 bar to 0.1 megapascal or 30 megapascal to 600. Say the lambda maximum, the emission maximum is changing at around 300 megapascal. It reaches the 355, above 350 nanometer. So that means the tryptophan side chains are in polar environment, or water is in contact with. All the tryptophans are fully hydrated at that rate. This is our that. So if we stop at this point, we conclude that the protein underwent a complete unfolding, and all the tryptophans are exposed to airborne water. But when we did a variable high pressure NMR, here is an array of proton NMR spectra as a function of pressure. We can see that even at the 300 megapascal, protein is in folded state. There are characteristic peaks coming from the folded state of this protein. This protein is in folded state, but all the tryptophan side chains, which are occupying the core region of this protein, they are hydrated. So using NMR, protein is in folded state at 300 megapascal. So when we combine these two, fluorescence and NMR, we can say that there is an extensively hydrated, there is a conformation state in which the core region of this protein is hydrated, or the water molecules are occupying the interior. So the equilibrium is shifting. So under normal pressure, the population of this conformation with the water molecules inside is very low. When we apply pressure, we are shifting the equilibrium towards this conformation state, which is in which the water molecules are occupied, or the interior of this protein is hydrated. So the protein is in the folded state, but the lambda maximum, the tryptophan fluorescence, shifting to 355 nanometer. This can happen. Only water is occupied or penetrates into the interior of this protein. So we proved the existence of an extensively hydrated, in which the interior of the protein is hydrated. Conformation is existing, which we call it as N prime. So we have a basic folded state N, and it is less hydrated, and another state N prime, an extensively hydrated state, and they are in equilibrium, and we are shifting our equilibrium towards N prime state by the application of pressure. So the tryptophan side chains, which are occupying the core region, and they are getting hydrated. This is the reason why lambda maximum was shifted to 350. So another important observation, we have different types of chemical shift changes, nonlinear types of chemical shift changes. As I mentioned earlier at the beginning, these kind of nonlinear chemical shift changes indicate the conformational transitions from one state to the other. Okay. So then we have another protein also there, the cavity filled mutant, V103L. So in this case, the tryptophan fluorescence, say even at 600 megapascal, it's around the lambda maximum is only 346. It's not going up to 355. That means the tryptophan side chains are not fully hydrated, even at very high pressure. So this cavity, so we have two proteins with one protein with a large internal cavity and the other one is with a small internal cavity. So this large internal cavity has some important role in the existence of this extensively hydrated and very dynamic. Then when we see the high pressure at the NMR spectra at 3000 bar or 300 megapascal, we can see that the lines are very broad. That means it is fluctuating. So the internal cavity is intimately related to the existence of the extensively hydrated power state in CMIP. So we can estimate the free energy difference between N and N prime by fitting a thermodynamic equations. So coming to the conclusion, the apparent discrepancy between the variable high pressure tryptophan fluorescence and variable pressure proton NMR results shows the presence of an extensively hydrated folded state in globular protein semi-receptor. The basic folded state L of CMM-R2 is in equilibrium with an extensively hydrated folded state in prime. The internal cavity is responsible for the existence of this N prime state in R2. The N prime state is more heterogeneous than, means it is more dynamic than N state. An interpretation of the conformation state of a protein based on tryptophan fluorescence alone may not be complete. But a combination of NMR spectroscopy is more important. Okay. Then what is the biological implication? So this is the DNA binding part of this semi-protein. So when semi binds to DNA, this tryptophan 95 side chain moves into this cavity. So this movement requires a much fluctuation in the structure. So we speculate that the more fluctuated, extensively hydrated conform N prime is more likely to be gold. Okay. So okay, hope you all are enjoying.
Yeah, thank you, sir.
No, yeah, we are continuing. Eh?
Okay.
Is time is over? How much time I have?
Oh, sorry, sir. Actually, this I thought that reference you kept maybe this way. Sorry.
Okay, okay. So maybe 15 minutes I can.
Yeah, please go ahead, sir. No problem.
Okay, okay.
Yeah.
So we are coming to the next example, second example. So all the results are published in Anglia Vandic. And this is about an interesting protein, HRAS. And this HRAS protein, it is a GTPase, and it is involved in the transcription and translation, and also in the signaling within the cells. Cellular signal transcription. So, it has a functional cycle. We call it as RAS cycle. And this, say, it has two different forms. One is GTP bound form. And this GTP bound form is the active form. And it is interacting with the different effectors and giving signal to cells for cellular growth, proliferation, and difference. And when this GTP gets hydrolyzed, it will go to the inactive form, GDP form. Guanosine triphosphate, all of it. So inactivation is there. Inactivation is transformation from GTP bound form to GDP bound form. And the activation when GDP is replaced with. So during this RAS cycle, RAS protein is involved in or it is interacting with the different partners. For example, so in the activation stage, it interacts with the exchange factors. In the inactivation part, it interacts with the GTPase activating proteins. And also in its function, it interacts with the different RAS effectors through which it gives signals to the cell. So, so when we look into the RAS cycle, so we don't believe, or it's not true to believe that the single conformation is interacting with all the other partners. And we say, or we believe, or we prove that different conformation states are occupied, or there are different states for RAS protein, and each one is responsible for, or each one is involved in interaction with a particular partner. For example, the one conformation state is interacting with this exchange part, and the other one is used to interact with the GTPase activate, and different conformation states are used to interact with the receptor. And at least 8 conformation states are occupied in the equilibrium for RAS protein. Why this RAS protein is very important? So this is an oncogenic. In around 30% cancer is happening because of this protein. In its some mutation, for example, glycine 12 valine mutation. It converts this protein into a permanently active form, which is very dangerous. But in its normal form, you require this protein for cell growth, cell proliferation. So in this study, we identified 4 different conformations for the GTP bound form. And we can target each conformation with small molecules. We call them drugs. And we can model, we can control this dangerous disease, this cancer. So coming to the experimental part. So we used the wild type RAS N15 labeled. So instead of GTP, we used a stable, GTP is hybridizable, a stable analog of GTP, GTP NHP. And magnesium is required for the binding of GTP to this protein. And we used this sample for NMR studies. And the spectrometer which we used was 800 megahertz NMR spectrometer. And we recorded different HSQCs, proton N15 HSQCs, as a function of pressure, up to 180 megapascal. So we can see different crossfits. They are changing as a function of pressure. And the temperature used was 302. Okay. Then we analyzed the data, the NMR crossfits, the positions. Position change in position means chemical shift changes as a function of pressure, and cross-peak volumes. Cross-peak volume change means population changes. So we analyzed all the cross-peaks which we could observe. Some of them were not appearing or they were overlooked. The observed peaks were assigned and placed. And then we determined the linear and nonlinear coefficients. The large P1 and P2, P1 and P2, different type groups we use different oscillations. D1 is linear coefficient, P2 is nonlinear coefficient. Large values of P1 and P2 indicate that for a particular residue, P2 is higher, is undergoing particular transition to one state. If B1 is higher, we can say that there will be fluctuations within this area. So we determined the B1 and B2 values of all the residues for both proton and neutron. And we plotted like this. It's important. So we color coded the residues on the three-dimensional structure of this RAS protein. The residues with the significant B1 and B2 values are color coded. Okay, different color codes. So we observed the volume changes also. The volume or intensity changes also. We can see that many residues are volume changes. And the residues showing significant, very significant volume changes. They are plotted from the chemical strategy. And different types of chemical shift changes and volume changes, plots as a function of pressure. Representatives, two-state, three-state, all these things. And we have different thermodynamic equations which we can fit to this and estimate the different thermodynamic parameters, delta G0, delta V0. We observe 4 states corresponding to these transitions. C2 is the most populated one at normal pressure, and C2 can undergo transition to C1 by the application of pressure, C2 to C3, C2 to C4, and different delta G0 values, corresponding delta G0 values, delta V0 values are shown here. And we identified the residues which are responsible for different types of transitions. Yellow colored residues are here, C2 to C1 transition, and then orange color, C2 to C3, and pink C2 to C4, then red, some residue sense, they undergo simultaneous transitions and different colors. So the population of each conformation state is changing as a function of pressure, like this. So we identify these 4 states: 3T state, 2T state, 1T, and 0. And this 2T state is responsible for the interaction with the effectors. 3T state is believed to be involved in the interaction with the adenosine activation, and then GTPase activity. And this 1T state is with the exchange effectors. And this is an intermediate one to which no nucleotide is attached. 1,0. So we identified this of this binds to the 1T state, 1T or C1 state. So, and this 1T or C1 state is believed to be involved in the interaction with the exchange factor. So here it is getting activated. The protein will become dangerous or cancerous when it is permanently activated. So when it binds to this state, it can regulate the activation, decrease the GEF mediate activation. Okay, this is the significance of this study. So we observe 4 functionally important conformation states in HRAS protein using efficient. So when a protein interacts with the different proteins in a function cycle, different structural states are required for the specific recognition of the patterns. In general, these structural differences are not restricted to the interaction site itself, as our hyperstring data nicely shows for activated. And these structural differences within or outside the protein-protein interaction surface create intrinsic allosteric binding sites that can potentially be recognized selectively by small limits. In summary, this non-classical type of allosteric interfaces provides a general applicable strategy for the modulation of the protein-protein interactions by small compounds independent of existence of transit level. allostery regulatory sites. So one more example is there. I will finish in 5 minutes. So there is another important protein called E. coli dihydrofolate protectase. And this enzyme, it reduces dihydrofolate into tetrahydrofolate, which is used for the synthesis of urine supplements. So we identified different conformational states in this dihydrofolate. We used the dihydrofolate from bacteria E. coli. So we identified two different conformational states, open and included. So this is the catalytic cycle of the DHM. The dihydrofolate is changing to tetrahydrofolate. And this is the three-dimensional structure of this protein. And it has 159 amino acids, 4 alpha helices, 8 beta sheets, 4 loops, including different residues, 6 to 23 methionine to the loop. This M20 loop, the loop formed by residues from 6 to 25. It's very important. So this is the NADPH. This is the cofactor. So this is the cofactor binding site here. So this, if there is no NADPH, most of the time, this M20 loop is occupying this NADPH binding site. We call it as the occluded conformation. So NADPH, the cofactor can bind to only when M20 is away from it. We call it as open conformation. So we identified using hypershell the conformational equilibrium between occluded and open conformation states. So here it is very clearly shown. This is M20 loop. So in the occluded form, this is the binary complex, a complex between E. coli DHFR and our substrate product. So in this binary complex, there is no cofactor. So this M20 loop can occupy this NADPH binding site. This conformation we call occluded form. And when it is away from this M20 cofactor binding site, we call it as open form. So we recorded different pressure and HSQCs of this protein using N50 label protein. And we observed that different residues are splitting as a function of pressure. So when we look into the structure, the location, the position of these residues undergoing splitting as a function of pressure. It's around the NADPH binding site. So this splitting indicates the equilibrium shift from occluded form to open form. So at a normal pressure, the occluded form is around, the population of the occluded form is around 90%. And the population of the open form is 10%. So when we apply pressure, the equilibrium will shift towards this open, which was a lower partial volume, and we can identify it. Okay, characters. So there is another interesting thing. How mutation, a single mutation, in a site which is very far from the active site, can affect the population of this open and occluded conformation. So so evolution happens for mutation mainly around the surface, for example, the surface loops. So we prepared a glycine 65 valine mutant of E. coli DHF. Here is the mutation site, G657B. Then we did all these variable hypershell NMR experiments. Then we observed the peak splitting as a function of pressure. The same residues around the NADPH units. And we plotted the changes in the peak areas or peak intensity or peak volume of the original peak and the new peak as a function. Sure. And then we estimated the delta G and delta G0. So initially, for the wild type protein, the population of the open conformation at normal pressure was 10%. And a single mutation, which is very far away from the active site, it changed the population from 10 to 18, almost doubled. So the population of open conformation almost doubled by glycine cystis E mutation. So variable high pressure NMR is a very important tool for studying the conformation of fluctuations in molecules, particularly in proteins, or the dynamics of molecules or dynamics of proteins. So the dynamics of a protein is very important for its function. So it is very key, it is very important to understand the conformation fluctuations. So variable high pressure NMR is a very good tool. So the groups with which I was associated for doing these high pressure NMR experiments. So initially I was with Professor Katsuyuki Akasaka at King University. Then I moved to this high pressure group of Professor Hans-Robert Kalbitzer. Then I was associated with this Kyoto Prefecture University group, Professor Mitsuki Oda. So I thank all the members of IRB's MG University. Again, I thank all the organizers, all the members of the Department of Chemistry, NIT Calicut. So all of them are very special for me. I'm very close to them because after coming back to India from Germany, I worked as an ad hoc faculty in the Department of Chemistry, NIT Calicut. I thank all of them for inviting me and giving me an opportunity to listen a different. I think this is a new technique to most of you. Okay, thank you.
Yeah, thank you, sir. Such a wonderful talk. Really very, very interesting, and how we can do the high pressure NMR also, what can be understand with high pressure NMR and this kind of protein folding and all. I think people are understood. It's really wonderful talk. Talk is finished. Now open for discussion. Audience, if you have any queries, questions, you can raise it. No? Okay. So I have one question. So when you bring this high pressure NMR, so what kind of NMR tube you are using?
Yeah, we use ceramic cell, ceramic.
Ceramic tube.
Yeah, we use a tube made up of ceramic material.
Okay.
It can withstand very high pressure.
Yeah. And one more doubt, actually, how we can pressurize that only the NMR tube? Like if when the NMR sample is inserted, then we can do pressurization.
Yeah,
during the
Shall I go to the one slide?
yeah.
There is a picture showing pressure
Yeah,
line.
maybe I missed actually that one. That's why.
Okay.
Uh,
Yeah, this is the picture of the high pressure setup at the
okay.
University of Regensburg. So we have a pressure generator.
Yeah.
This pressure generator can be either manual one or an automatic one.
Okay.
The difference is that for changing the manual one, we have to change the pressure manually for rotating
Yeah.
this piston. But in the case of this automatic generator, we can control the pressure using computer programs. We can
Okay.
set all the pressure points. It will automatically change. Okay. Then uh, there is a pressure line.
Yes.
Okay. And this pressure line is connected to this NMR sample tube.
Okay.
Okay. And we have a pressure mediator. Usually we use water as the pressure mediator for
Okay.
transmitting pressure from the pressure generator to the sample. And we
Yeah.
have to separate the sample from the water, the pressure mediator. For separating them, we use a membrane,
Okay.
an elastic membrane. Okay. So this way we can transmit our pressure to the sample.
Okay.
Okay.
Yeah. So for this one, we need to use separate probe for this.
No, normal probe.
Normal probe. Normal
Yeah.
probe is fine.
Yeah. And in addition to this, we have a safety jacket also
Yeah.
around this tube. Okay. If it breaks, then everything will be there in the safety jacket.
Okay, and one more final question. Actually, you work only on the proteins. Mostly, we can see the protein, how the structure changing proteins and all.
Yeah.
It's like a small molecule. Also, we can do
Yeah,
this.
we can do that.
Yeah.
So there was so I forgot the name of that catalyst. I did it many years back. A small molecule which is working as a catalyst. I forgot the details.
Okay.
So
Okay.
it was a work in association with the chemistry department of University of Regensburg.
Okay.
So at higher pressure, this molecule, this catalyst is showing much catalytic activity.
Yeah.
Then these people didn't know why.
Okay.
Then they came to us, and
Okay.
we checked the conformational change.
Okay.
What is happening as a function of pressure?
Yeah.
Then we observed that this molecule, the population of the active form, the functional form, is increasing with pressure.
Okay.
That is why it was showing higher catalytic activity. So we can apply pressure
Okay.
to small.
Okay. So actually, this is like a generally the mechanical force when you are applying the pressure. Like we can detect very easily by fluorescence. Like we can easily understand. As you know, this fluorescence is very sensitive. So how much nicely we can see in NMR when you are using the pressure. Like we need to use like a much high pressure. As you know, fluorescence we no need to use much high pressure.
You mean the pressure level which we have to use
Yes.
in a particular protein system, you mean, right?
Yes.
Okay. So it varies with the protein, the pressure range which we have to use. In both of the case, fluorescence as well as with the NMR. So we can do different types of studies using high pressure. One thing is fluctuations within the faulted states.
Yeah.
So in that case, first we have to optimize the pressure limit, because after particular pressure value, the protein will completely, the equilibrium will shift completely to the unfolded state. But we need to study the folded, the conformational fluctuations or conformational equilibrium within the folded state. So first we will optimize or we will find out the pressure limit below which the protein is in the folded state and above which it will go to unfolded state.
Okay.
And suppose we need to study the unfolding of protein.
Okay.
Then we have to go to higher pressure.
Okay.
Irrespective of the technique which we use. The
Yeah.
fluorescence and NMR, both we are using just to observe what is happening to the.
Okay. 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 Thank you very much. Really such a wonderful talk. And anybody have queries? Audience? Okay. If nobody have any queries, and we'll join thanks to Dr. Sunil Kumar, such a wonderful talk. He showed very interesting systems, how we can modulate the folding and anti-folding of proteins extensively. His work is, we can see it's how nicely he showed. He talk also. It's a very good. Thank you very much, sir. And hope
you. Thank
see once maybe the corona down, maybe we will invite you to visit our institute. Then we can have a very nice meeting. And thank you very much, sir. Really appreciate.
Thank you very much.
Yeah.
Okay.
The mic transfer to the room, committee room. Then you can talk. Hello,
Yeah.
Sriya?
Hello.
Yes, sir.
Yeah, please go ahead.
Thank you so much, sir, for the informative session on high pressure NMR. Thank you so much.
Thank you.
And moving on to the second session of the day, our respected speaker, Dr. S. Prabhakar from CSIR IICT, will give a lecture on the topic Mass Spectrometry Principles and Recent Advances for Structural Characterization. I cordially invite Dr. Lakshmi C., Professor, Department of Chemistry, NIT Calicut, to chair the session. Over to you, ma'am.
Good morning, everyone. It's my pleasure to invite Dr. S. Prabhakar from IICT Hyderabad. I'm also a student from there. I did my PhD there, so it gives me immense pleasure in doing this introduction. So Dr. Prabhakar completed his BSc and MSc from Osmania University, and later he joined CSIR IIC degree at the mass spectrometry facility there. And after his PhD, he continued there as a research associate, and subsequently he became a permanent staff, permanent scientist at that facility. And he is continuing there as the senior principal scientist at the Mass Spectrometry Center of IICT Hyderabad. And his expertise is in the analytical chemistry, particularly the work related to the mass spectrometry. And he mainly does the structural characterization of small molecules, natural products, and biomolecules, etc. And the application of mass spectrometry to the basic and applied research on metabolomics, metabolite profiling, isomeric differentiation, to name a few. And he has published extensively in top journals like chemical communication and several biology journals. So, and apart from that, he also received several awards and honors, like eminent mass spectrometrist in 2013 by ISMAS, and fellow of Telangana Academy of Science, CSIR IICT Gaurav Samman Award, etc., to name a few. And I have a personal connection with that mass spectrometry unit there, because when I was a PhD student there, I used to visit that facility on a regular basis, and I still remember the smiling face of Dr. Vairamani sir and his student and his team, who always supported us without any with all the support they extended during my stay there. So it's gives me so much pleasure to welcome someone from the same facility to this workshop. So, Prabhakaran sir, it's a pleasure. So, we cordially welcome you to the session. Hello, sir. Was I audible?
Yes, ma'am. Yes, ma'am. You're audible.
Okay, okay. Sir?
Yeah, am I audible, Lakshmi madam?
Yes, you are, you are.
Okay.
Okay.
So good morning, everyone. It's my pleasure to give a lecture on this one. So just now I happen to know that Dr. Lakshmi, she has done PhD from IICT. So nice to hear you, Madam Lakshmi madam.
I think we met before. I remember your face. We met before.
Yeah, okay.
Yeah.
So what time you did your PhD?
I was there from 98 to 2001, and I worked with Dr. Darshan Ranganathan and Subramania Ranganathan. So I was a regular
Oh, okay.
visitor at your facility. Yes.
He is a guru of my guru. So
Yeah, I know.
yes, yeah. Yeah, so nice to meet you, madam.
Yeah.
So
Nice to meet
first,
you.
I would like to thank Dr. Anuj and Dr. Lakshmi to give me this opportunity to speak about mass spectrometry. Madam, can you confirm whether my slides are visible?
Yes, it is. It is visible. Yes, sir. I can see it.
Yeah, it's in the full screen, right?
Yes.
Thank you. Thank you for confirmation. So when should I close, madam? By 1 or 11, 12:45? I can go up to 12:45.
Yes, sir. It can take some more time. Also, 1 o'clock. Up to 1 o'clock, it can take, sir. No problem.
Sure. Thank you. Thank you for confirmation. So there is no break in between. So I hope you are all okay. So my lecture will be on mass spectrometry. I will cover basic principle and recent advances. And Dr. Anuj also mentioned that cover HRMS. So I will be covering what is HRMS and what is the use of it. So the overview will be, I will start with basic mass spectrometry because in order to understand the interpretation of mass spec, you need to know how to calculate molecular weight and what is HRMS, what is isotope, what is isobar. So these are all required. So I'll be covering this one. And also I'll give some examples at the end and some important fragmentations also I will cover at the end. So if you see any researcher, they will be pointing at some point. Okay. For example, physicists, they look at electron level or neutron level or positron level. So they normally look at atom and further, but we chemists, we look at molecules. Okay. And whereas the biologists, they look at the cells and other things. But whenever it comes to the mass spectrometry or any structural characterization, so we will be looking at molecular level. So you should know what is a molecule and how it is formed by bonding with atoms and those things. Okay. So basically, as an analyst, we'll be looking at what is there in it, like a qualitative analysis, and how much is there is like a quantitative analysis. So everybody looking at as one of these questions. So if it is a mixture, you will be looking at like qualitatively what kind of molecules are there, and if you want to know the levels, then you go for quantitative. So both the things can be done by mass spectrometry. So as I said, now we are looking at molecule level. So obviously we cannot see with an eye and we cannot handle the molecules with our hands. So individual molecule, I mean. So if you see, by eye we can see maximum 0.1 millimeter, and even if we use a microscope, it will be 0.001. So if you see this graph and the red line, whatever is there, left hand side you can see your hand, so you can handle with microscopes and other things. But right hand side you have nanomaterials and small molecules and atoms, proteins, peptides, virus, like this, which you cannot see. So that's why you need analytical equipment, various analytical equipment to characterize these molecules. So if you look at characterizing molecules, as you know, so there are like spectroscopic techniques where UV, visible IR, and NMR just now finished today. There you can get structure information in NMR, but UV and visible IR, you get functional groups or no, like partial information. It may not give the full structure, whereas NMR can give, but sometimes it may have some difficult to confirm the molecular weight and elemental composition, and quantitation also may be difficult. You require a little higher amount of samples. So in that case, now we need a mass spectrometry technique. Thing is, it is not a spectroscopy technique because there is no involvement of electromagnetic radiation, either absorption or emission. Whereas, if you see UV, IR, NMR, there is an interaction of electromagnetic radiation, either absorption or emission. But when it comes to the mass spectrometry, I will cover in the basics next. So there is no involvement of electromagnetic radiation. That's why it is called a spectrometry technique. Okay, so the instrument is it is called spectrometry. Only thing is it will be in the gas phase, and this technique is a destructive technique. That means once the sample enters into the instrument, you cannot retrieve it, and it is a non-additive. Only a molecular level you can look at. So if you want to see any nomenclature, there are many books for nomenclature, and there is one recent IUPAC one also, glossary also is there. Those who wanted to know more about definitions and those, you may look at this one, or at least download the language of mass spectrometry. It was appeared in Pure and Applied Chemistry 2013. So there the authors discuss what are the new definitions, and this nomenclature only we have to use. After 2013, the nomenclature is changed. When you write your paper or when you write whatever thesis, you should make sure you use this language. You can download this IUPAC journal paper. Okay, so to brief you, there's one or two terminologies which were changed. Some of them I listed here. You can see later also. So the first one is the electron impact. Earlier you used to say that is the first mass spectrometer where you do. I will cover one slide on EA also. Okay. So now there is no more electron impact. So you have to use electron ionization. So like that, earlier in the case of LCMS or FAB, there is a proton is added to the molecule during ionization. In that case, earlier we used to say protonated molecular ion and pseudo molecular ion. Those are gone. Now you have to say if it is a proton is added, you have to say protonated molecule. If it is a sodium is added, you can say sodiated molecule, like that. Okay. So like that, MSMS. Yesterday, I think yesterday or day before, my colleague might have given. So there also product ion scan, precursor ion scan, multiple reaction monitoring. So these are the new terminology. Earlier we used to say daughter ion, parent ion. So there was a conflict when you say daughter or son, something like that. So this terminology you have to be careful when you are using now. So as I said, these are some of the journals one can look at, selected only with mass spectrometry. So if you want to read some books, these are some good books available. Mass spectra of organic. So these are all books. So I'm not going to fully cover the history of mass spectrometry, but you can understand when the mass spectrometry started with J.J. Thomson, and then Aston is the one who made the mass spectrometer. He started using for atoms. As I said, it was mostly physics people. They were looking at atomic structure, atomic molecular weight, like that. All periodic table atomic weights, most of them were covered by mass spectrometry, isotopes and isobars and those stuff. So after that, then Bannon and this one, no, the water pole analyzers have come. Then you got GCMS and LCMS later on. So now there is no limitation of mass spectrometry. You can start with hydrogen and you can end like protein, peptide, virus, whatnot. The technology is improved and there is no limits for the mass spectrometry applications. Any molecular weight like so far 3.5 kilodalton or even 500 kilodalton. So these are the noble laureates in mass spectrometry. As I said, the first one is the father of mass spectrometry, J.J. Thomson. Then Aston made the first mass spectrometer. But if you see the right hand side, the Fenn and Tanaka, they were the people. Actually, they showed the application of mass spectrometry to the biology. Whatever we have now, electrospray, MALDI, these techniques, there are because of these two, and that's why they got the Nobel Prize in 2002. Actually, they shared the Nobel Prize. So as I said, even for mass spectrometrist, because this is the one which gives you the atomic weight, molecular weight. This is the basic for molecular weight calculation. And if you look at the values given there, the top one is the atomic mass and the bottom is the atomic, the top is atomic number and the bottom is the atomic mass. So why I'm telling these means, unless you get the correct atomic mass, then only you can calculate molecular mass. Okay, so I'll give you some definitions. If you don't understand that one, you cannot interpret the mass spectrum. So this is the how you calculate molecular weight. So first you need to have atomic weight. So atomic weight is nothing but number of protons and number of neutrons. You combine them, and molecular weight is a sum of atomic weights, whatever elements present in the molecule. Those atomic weight you combine to get the molecular weight. Of course, electron is too small. That's why people don't consider the number of electrons, but maybe high resolution power, if the instrument is able to cover fourth decimal accurately, then it is important to consider electrons also, but nowadays not important at this moment. So as I said, the molecular weight is not too high. You cannot measure in milligram or nanogram, femtogram like that. So there are some units you need to follow as per new IUPAC. You have to either unified atomic mass unit, U, or Dalton, you have to say. So there are no other units you have to use, only these two. So you must be knowing what is an isotope and what is an isobar. So these two are very important for the mass spectrometry. So isotope means same atomic number but different mass number. So there is a slight change in the number of neutrons. For example, hydrogen, you have hydrogen, deuterium, and tritium, and there is a change in neutrons. I will not go into the details, but isobars are different atomic number but same mass number. That means when you get into the mass spectrometer low resolution, isobars you cannot differentiate them because they have the same mass number. Whereas isotopes, yes, you can do it because they have different mass numbers. So this you should remember. But if you have a high resolution mass spectrometer, to some extent, depending on the resolving power of the mass spectrometer, you can differentiate isobars because the accurate mass will be different. I will tell you what is an accurate mass. So as I said, we are actually calculating the molecular weight. So then spectrometer, because actually it is a mass value we are getting. Mass is nothing but it is a constant value, and when it comes to the weight, as you know, the gravitational force comes into the picture. So in mass spectrometry, there is a vacuum, and there is no effect of gravitational force. You will be getting actually the mass value. So that's why it is called mass spectrometer. Okay. So if you look at, so this I already explained you. Why people are looking at mass spectrometer? The number one, it is a speed to get one mass spectrum instrument, and it is highly sensitive. Even now, we are claiming like PPT level, parts per trillion level, we are measuring, and it is very specific. There is no like a molecular weight 100 and 101 means very specific. There is no question of plus or minus 1. Okay, highly specific, accurate mass. And the other thing is you can couple mass spectrometer to the either any chromatography technique. Here I have given some examples. It can be coupled to GC, it can be coupled to HPLC, it can be coupled to capillary electrophoresis, even HPTLC. Also you can couple. So we have, so when we couple to chromatography technique, it is called hyphenated technique, like GCMS, LCMS, CEMS, ICP-MS, whatnot. You have many hyphenated mass spectrometry techniques now. Okay, so if you look at the advantages with mass spectrometry, as I said, the primary thing is the molecular weight. Yes, but if you have a resolution mass spectrometer, then you have additional information, elemental composition you can get. That is additional. And if you have the MSMS capability, then you can get structural characterization, because you'll be breaking the molecule and each fragment will give some information, then you can stitch together, you get the final molecular weight information. But if you have high resolution MSMS, then additional fragment and also you get elemental composition. So like that, nowadays, the characterization of structure of small molecules and even peptides, proteins, it is made easy because of this MSMS, that too with high resolution. So mixtures, obviously you can analyze mixtures and you can do quantification. The only limitation is whatever molecules you are looking at, it has to be brought into the gas phase. If you cannot ionize, then you cannot analyze by mass spectrometry. That is the only limit, but we have very high-fi techniques, to bring the very high molecular weight compounds in the gas phase. Only other thing is the sample cannot be retrieved. But anyway, you are introducing like a very femto or micro gram level, not even micro, femtogram. So as I said, if you look at only GC or LC is a two-dimensional data where you get retention time and the intensity. That is the two values you get. But once you connect to the mass spectrometer, you have a third one, the spectrum of each peak. Each peak, in the sense, one peak can have like 10 to 20 spectra or even more. Okay? You can average and get the mass spectrum. So each peak will give a third dimensional data mass spectrum. And if you have an MSMS, then you have a fourth dimensional data where you get the fragment ends. So that is the advantage of either GC-MSMS or LC-MSMS. You have a third dimensional, fourth dimensional data where you can get structure information very easily. So now let us come to the how you get this spectra, what mass value you look at. Okay, so there are, as a definition, you have as long as the mass spectrometer separates the isotopes, you have to look at monoisotopic mass value. How do you calculate monoisotopic mass value? So you use atomic mass of the most abundant isotope. For example, carbon 12, fluorine 35. So like that. So you use most abundant isotope and you get the molecular weight. But as the molecular weight increases, I can say more than 5,000 Daltons, 6,000 Daltons, then there may be a difficulty in separating isotopes like proteins, big proteins, peptides, big peptides. There, because isotopes may not be separated, you have to go to average mass. But as long as small molecules in up to 2,000, 3,000, 4,000, no issue, you get the isotopes. You have to look at only monoisotopic mass value. Okay, that means whatever you get in the periodic table, whatever you get on the bottle of your compound, whenever you buy, there is an average mass value given on the bottle. So when you record the spectrum, don't look at those average mass value. So please look at, nowadays softwares are there. How do you calculate isotopic mass? Just Google it, it will come. You put your formula, it will give you monoisotopic mass value. Okay. So please look at the monoisotopic mass value to interpret your mass spectrum. But when it comes to the high resolution, then you have another two definitions. Okay. So normally in the low resolution, we don't measure the molecular weight in the decimal value. So that's why you actually get the nominal mass. That means you are rounding to the nearest integer value. So you know what is the nearest integer value. If it crosses 0.5, then you go to the next integer value. Okay. If it is below 0.5, it will come to the left. So that is the nominal mass. So either average mass or monoisotopic mass can be nominal mass if you are using low resolution instrument. But when it comes to the high resolution instrument, then you will be using the mass in decimal places. Okay. Then there are two definitions comes into the picture. One is the exact mass, the other one is accurate mass. Okay. The exact mass is the calculated mass from the every atom has a exact mass value and it is fixed. You can just combine all exact masses of atoms present in your molecule, then you get the calculated value. So that calculated value is exact mass, it is fixed, never changes. But when you measure the mass value in decimals by using mass spectrometer, that is called accurate mass. Okay. That accurate mass is nothing but an experimentally determined mass. Okay. So you should not confuse between exact mass and accurate mass. Okay. If you get a HRMS spectrum, you have to say the accurate mass determined by the instrument was this. You should not write the exact mass was you. Who are you to measure the exact mass? The exact mass is already there in the standard. Okay. So please remember these 2-3 definitions in order to understand the high resolution. Anyway, you get in the literature the exact masses and nominal masses of common elements and isotopes. Even there are you can browse Google. It will give you if you want exact mass, it can calculate for you. So this is the introduction. Then now let me explain you what is the mass spectrometer and the parts. I will very quickly go because most of you may be knowing, but there will be 3 parts in mass spectrometer. First one is the source where you produce the ions, and the second one is the analyzer where you separate ions. And if you have MSMS, then you have more than one analyzers. There will be one source, one detector in the MSMS instruments. But only difference is you will have more than one analyzer and detection of ions at detector, and the entire part will be under vacuum. Okay. So why under vacuum? Because you are generating highly reactive species in the gas phase, because ions are not stable in the gas phase. They undergo ion molecule reactions. That's why vacuum is essential part in order to make them live and till it detects at the detector. So higher the, so nowadays we have like a 10 to the power of minus 8 torr or minus 9 torr in the analyzer and detector. But now new techniques like LCMS, we have the source part in the normal atmospheric pressure, but once ion is formed, it will go to the vacuum place. Okay, it will not be disturbed. These are overall parts of mass spectrometry. As I said, first place the gas phase ion generation where ions are formed. In the some cases, this source is in the atmospheric pressure, like LCMS. But whereas the analyzer and detector are everything under vacuum. And finally, at the detector, you detect the current of the ion, then you get the time versus abundance spectrum. Then you calibrate the instrument with a known sample that will convert to mass to abundance. So that means the calibration of the instrument also very important. Okay. Then the question comes when to calibrate the instrument. Most of the high resolution instrument you have to calibrate daily or at least once in a week depending on the analyzer. But most of the instruments are daily calibration for a high resolution. So if you have a low resolution quarter pole instrument, years together, no issue, 6 months or 1 year, the calibration will be there. But you should remember, unless the calibration is done correctly, you cannot get the mass value correct. So the mass spectrometer measures the molecular weight. This is the statement normally we give, but if you really think is actually mass spectrometer measuring not the mass value, it is the mass to charge ratio in the gas phase. That is the actual measurement of by mass spectrometer. So this is a spectrum just to show you. It is a EI mass spectrum. This is a plot of mass to charge ratio and the relative abundance on the y-axis. Because the molecular ion EI, because of EI, it showed many fragment ions. I'll tell you in the later stages. So extreme right one is a molecular ion. If it is stable, then you got the highest ion, which is base peak, and in between you got the fragment ion. This is how the spectrum looks like in GCMS, like EI. But if it is a LCMS, that one you get only single peak, less fragmentation pattern because of the soft ionization. This I'll skip it. Yeah, already explained you. If you see the, as I said, first part is the ionization part. In GCMS and EI, you are playing with electrons. That is the top one. There are only two types of ionization. One is the playing with electrons, where either you can add electron to the molecule or you can remove electron from the molecule. That's what in electron ionization. If it removes one electron from molecule, it is positive ion. If it adds one electron from to the molecule, you get the negative ion. This is the common thing. This is the first ionization technique in mass spectrometry. But now we have a latest techniques like fast atom bombardment, electrospray ionization, APCA, APPA in LCMS, MALDI. Here in the soft ionization, you are actually adding a cation to the cation or a proton to the molecule. You get M plus H, M plus Na, M plus K, M plus ammonia, depending on the mobile phase in the LCMS. It will change. But if you see the ionization part, actually you are adding a cation to the molecule to get a protonated, sodiated, or potassiated like that. In negative ion mode, either deprotonated or you can add a negative ion to the molecule to get negative part. Okay, so this is how the entire mass spectrometry organic molecules or proteins, peptides. You get protonation. In the case of course, in proteins, peptides, you get multiply charge in electrospray. that is a different issue. But overall ionization is one of these two, okay? Either electron or proton. So this I just try to summarize what actually you get in different ionization techniques. Anyway, it's understood. Here I don't explain you, but you know already what is electron ionization. Is the first thing you learn in your. It's like electron bombardment. You get fragmentation because you are using more than 70 electron volts. Actually, to remove electron, 15 to 20 electron volts, because you are using 70 electron volts, so it will fragment. But you need fragmentation for structural isolation. That is another aiming for the EI analysis. So just fragmentation. There are many homolytic, heterolytic fragmentation, rearrangements. Everything happens in electron ionization. Okay. You should be careful when you are interpreting the data. Are you looking at EIMA, GCMS? In the case of GCMS, you have to look at this fragmentation patterns, and if it is a soft ionization LCMS, you get only protonated, associated, or deprotonated molecules. As you know, sometimes electron ionization cannot give a molecular weight, so you have to go for chemical ionization where same process, but you introduce a Ca reagent gas, where the electron beam first ionizes the Ca reagent gas, and those reagent ions will interact with your molecule to get the M plus H ion. Everything happens in the source, chemical ionization source. So there are like methane Ca where you get protonated, and isobutane Ca. There are 2-3 reagent gases. Anyway, this is a very well-known technique. But just one example. If you miss, there is one compound, ephedrine. Everyone knows what is an ephedrine. So in EI mass heat case, only one ion, 58, a small fragment ion, which is just gives, it will not give a full structure information. But when you do the chemical ionization, then you can get M plus H ion, then small fragments, everything. So those who are handling GCMS, then this is required. Ca is required for the molecular weight confirmation. So when it comes to the LCMS, then you have 3 different ionization techniques. One is the electrospray ionization, where you send your solution either from HPLC or directly also you can insert. The solution goes through a capillary which is at high voltage. Then other side you get the spray, that is called electrospray. Spray is nothing but charged droplets. Okay, by using nitrogen you remove the solvent. It is called a desalvation process. Then the ion is free from the solvent. It will go to analyzer. So that's how the electrospray, you can see here. This is the droplet you get where ions are there, and remove the solvent by nitrogen pressure, either hot nitrogen or flow of nitrogen. Then it will reduce the size. As the size is decreasing, then you cannot accommodate so many ions in one small size. Then it will explode to give an individual ion. That is the Rayleigh limit we say in physics. So that's how you get gas phase ion, then goes to the analyzer. So there's the electrospray. Just one point I want to mention here. Whoever is having LCMA, there is certain voltage we give it to the other side. This is where the ions are going into the analyzer because the interface or inlet, MS inlet, okay? And the left hand side you have the electrospray. In between you are applying some voltage, okay? Because it has to pass from, it has passed from left to right, okay? Because for example, you are running in a positive ion mode, okay? In order to get positive ion to the inlet of MS, then you need small negative charge on the MS inlet. Okay, so the ions are dragged. Okay, but if you use a little high voltage on the MS inlet, then the ions will come very fast towards the MS inlet. In this process, it can undergo fragmentation. Okay, that means if you are using a little higher voltage on the cone or whatever MS inlet you say, if you that should be optimum. Okay? If you use a high voltage, then it will fragment. If you use a optimum voltage, then it will not fragment. So this process is called in-source fragmentation. But sometimes this is advantageous. If you don't have LC-MS-MS, LC-MS itself you can separate in HPLC and do the in-source fragmentation. At least you get some fragmentation pattern here to get the structure. This is another tip for having LCMS. You can do MSMS type of information. Okay. In fact, recently it was analyzed. It is appeared in 2021, very recently. So, what is the difference between source fragmentation and also real MSMS, where you select ion and do the MSMS? So, hardly they are very close, whether it is fragmenting in the source or whether you have done MSMS, it is almost close. So that's why whoever have a single quad instrument, they can actually attempt this one to fragment. But thing is, optimum voltage has to be used to get the actual MSMS fragment ions in the source. Okay. So using this technology, where by applying high voltage you get the electrospray, the recent techniques, they are like fancy paper spray. Okay, this is like you fold the paper in a triangle and you put your blood sample over it and you apply the voltage at one side. The other tip, you get the electrospray like thing. Okay, because it is spraying. Once it sprays, you apply nitrogen and you get the free ions, you get the spectrum. So without any LCMS, without any other thing, simply you are applying, only thing is the paper has to be wet. So before applying high voltage, you either apply water methanol or methanol, something. It should be wet so that the voltage will be applied to the paper. Very nice technology. And many blood samples, many applications have come using this paper spray. But when it recently, not recently, but it is there, similar technology where you can take live plant leaf. And as you know, leaf has the cells and cell has the water. You don't have to wet it, what is already there. So sometimes you can use a methanol for extraction and you apply the voltage at one end. Other end, at the tip, you get the spray, you get the mass spectrum. This is called a leaf spray. So like that, many techniques are there for the electrospray. Okay. APCA is another source where you have the same capillary, but this time you use high temperature to get the sample vapor with solvent. Then you have a corona needle where high voltage is applied. Your solvent will ionize first and the solvent protonates your molecule by ion molecule reaction. It's like exactly chemical ionization only, but there is no reagent here. The reagent is the solvent itself acts as a reagent in APCA. In APPA, actually, there is another technique. I'm not going to detail, but I'm going to share these slides. In this case, it is similar to APCA, whereas instead of corona needle, you have the EV source, photon source. Okay? You bombard with 10 electron photons. The neutrals which have come, they get this photon energy and they will ionize. Either you get molecular ion, or because of the solvent, it will protonate. You get M plus H like that. Okay, this is other technique you can use in LCMS. So MALDI is other technique. I'm not going to details. Where no, this is actually MALDI is a solid state ionization, where matrix and sample are crystallized. I mean, you make a solution of both matrix and compound, and you evaporate the solvent. Your analyte will be co-crystallizing with your sample matrix, and then bombard with the laser, you get the MALDI. That is the matrix assisted laser desorption ionization techniques. This is mainly used for the high peptides and polymers, proteins, whatnot. Okay, the other very desi technique, dart technique. There are many applications, but it is similar to your electrospray, like that. Okay, there is I also developed one technique in the US. It's called laser ablation electrospray, where you keep cells in front of the mass spectrometer, then you bombard with a IR laser, and the water abstracts the IR laser, and cell is broken, and the neutral molecules will come up, and you do the electrospray. Just solvent you spray, and then you get the ionization. That will go to the analyzer. Ions will go to the analyzer. So like that, techniques are like many techniques are being developed for direct analysis. Any type of compounds you can analyze. This is another recent technique there in London. When the doctor operates the tissue, when he cuts the tissue, actually it will give some vapor, molecular vapor. Okay, and dynamically that vapor is abstracted from there and send it to the mass spectrometer. Okay, so whenever he is cutting, whether he is cutting malignant tissue or whether he is cutting the normal tissue, he can get it based on the spectrum what he gets on the screen. So this is called an intelligent knife. Like this, no fancy. In future, we may get such kind of equipments in the diagnostic labs, and I think you must be knowing many labs in Hyderabad. They have mass spectrometers. Now they are doing steroid test and your vitamin D metabolites, whatnot. Now it is being used as a equipment in hospitals. So TB patients, everything they have been used. It has been used for dynamic checking. Okay, so that's about the ionization of different things. So let's come to the analyzer part, where whether you have a low resolution or a high resolution. That part comes in the analyzer part. So let me just go through quickly. Magnet I am not going to touch. The water pool analyzer is the common low resolution analyzer, where 4 rods will be there, and I'm not going to the principles, but only thing is, if you apply very specific RF DC voltage, one ion will be filtering through. Okay, so that's why in MSMS this quarter pole is used to select the ion. Okay, so if you sweep the voltage, it can do full scan. If you fix the voltage, only one ion will go. So that is the advantage with water pole analyzer. But whereas the time of flight analyzer, this is another one where a flight tube will be there, and you send ions together, and like a running race, it will go in the flight tube. If it is light molecular weight, it will go faster. It will heavy molecular weight, it will go slow, one by one. That's why it is called time of flight analyzer. But the thing is that you have a linear TOF, a reflectron TOF. In the reflectron TOF, like a running race, you have a 100 meters, 500 meters, like Why you are going to 500 meters? Because 100 meters may not be sufficient to know who is coming first. Okay, like that in mass spectrometry also. There are like some molecules having very close arrival of the ion to the detector. So in that case, you have to increase the path so that it will be separated because of the increasing the path length. So like that, the reflectron TOF is there. You know, any MALDI TOF, Q TOF now we have, that is a high resolution instrument because of this reflectron tubes. You can use 2-3 reflectrons and you can, it is called a V tube, but you can also have a W tube nowadays. Ion cyclotrons also coming where the ions move in cyclotron way to increase the path length. This is one of the high resolution, and other high resolution instrument is FT. I think only 5-6 in the world because problem in taking care of this. But now the latest instrument is the Arbitrap analyzer, where this can give very high resolution instrument. And nowadays it is, earlier it was 1 lakh, but now we have 2,40,000 Arbitrap instrument. And it is cheaper when it compares to the other magnetic sector instrument. But here the technology is you send the set of your molecules inside the Arbitrap. There will be two, so centered electrode will be there where your ions are moving in a circular path, and each ion will have different circles so that they will be separated, and the moment the resonance it will get the detector. Okay. Anyway, but the thing is, Arbitrap analyzer is the current high resolution instrument where you can use small molecules. Only thing is the mass limit will be there. For TOF analyzer, there is no mass limit, but whereas the Arbitrap you have a mass limit. So detectors I don't bother because detector is just once the ion touches the detector, it will generate some current, but the current is not that much to measure. It's not measurable current. You multiply the current, that's why it is called electron multiplier. But when it comes to the TOF analyzer, normally we use a microchannel plate for the TOF analyzer because we have multiple electron multiplier like thing. Micro channels will be there. So it is highly sensitive detector. Wherever time of flight detector is, time of flight analyzer is there, MCP detector will be there. So if you see now, I covered some sources, some analyzers and detectors. So you can, depending on your usage, you can configure yourself what kind of instrument you want. You want whether GCMS or LCMS, you want low resolution or high resolution, like that you can select and buy your instrument. So it's like a pizza, what you buy, you customize your pizza like that. Nowadays, any combinations you can get it. You have to choose your source, choose your analyzer and detector. Give me 1 second. So because of the development in mass spectrometer, so earlier if you see those who are in 1990s, mass spectrometer means big hall will be there and you have a very big instrument like what is shown here in the left and top. So this is mass spectrometer, but because now analyzer changed and it is miniaturized, you can see GCMS now it is the mass spectrometer is less than the GC. Like you can keep your mass spectrometer in the swimming cupboard, or you can keep like a PDA. You can also have mass spectrometer on the top of HPLC. So like that, many developments are there. Now mass spectrometer is also sent to the space in Chandrayaan 1. They sent a mass spectrometer into the space. So we can imagine, no, mass spectrometer is like it can do wonders now. Okay, so I'll just quickly go through the importance of HRMS. Now you have heard about what is a mass spectrometer and what is a source analyzer detector. So as I said, when it comes to the high resolution, your aim is to get the mass value in the digits, so that you can get elemental composition. So if you see, the first thing is how the peak appears in the mass spectrum. Okay, but usually, like NMR, IR, UV, mass spectrum also, the peak will be in the Gaussian shape. Okay, the continuum spectrum given here, if you see, actual peak is like this. But only thing is, at the time of recording, you convert into the line spectrum and record, because this is only to avoid a number of data points. But if you want, you can also record the spectrum in the continuum mode, where it can take more data points. But what I mean to say here is, the actual peak is in the Gaussian shape. Okay, so this peak shape and peak width is important for high resolution. Okay, so high resolution is nothing but the power of the analyzer to separate the closely coming ions. Okay, whether it is separating 0.1 Dalton or not. So depending on the ability to separate, it is called resolving power, okay? Resolution. So let us see. In magnetic sectors, you have, if you have two peaks, and if two peaks are separated with a value of 10%, then the peak resolution would be m by delta m. Okay, the mass value of the first peak, and you take the mass difference between the two peaks, and you can calculate the resolution. Okay, that means if you want to separate 500 and 500.1, the resolution required by the instrument will be m by delta m. Okay, so what is the m value? 500. Delta m is 0.1. Okay, so 500 by 0.1, it will be 5000. That means the resolution of mass spectrometer should be 5000 to differentiate 500 and 501. Okay. So the another time of flight, if you have a time of flight, how do you calculate the resolution? The resolution is the mass value divided by the peak width at 50%. Okay. So here the peak width is 0.1 Dalton. Okay. Peak width at a half maximum, okay, at 50%. So same thing, if it is 0.1 Dalton peak width, then 500 divided by 0.1, you get 5000 resolution. So that's how you measure the resolution of the instrument. Okay, let us see some example here. Okay, either you can take delta m, and m by delta m will give you resolution if you have two peaks. If you have a single peak, then you should know the mass width at 50% to get the resolution. There are two ways to get the resolution. Okay? So once you have the resolution and you are getting accurate mass from the from the instrument, then the question comes, what is the error value? Because no, you have exact mass which is theoretical, okay, and you have the measured mass which is from the instrument. So if you want mass accuracy in ppm, then the embedded value what you measured minus embedded value which is theoretical, okay, exact mass, divided by embedded theoretical, so into 10 to the power of 6. So if you apply this formula, you get ppm value. But some people, they use MMU. In that case, M measured minus M theoretical, M measured into 10 to the power of 3. Okay. So this is a reference you can read. Normally, in any HRMS, we take in ppm, and it should be less than 5 ppm to get the accurate mass value. Then only it is acceptable, any HRMS. So here, depending on as the mass value increases, your required resolution will be increased. Okay. Here I am giving you one example where to differentiate 100 and 101, you require only 100 resolution. Okay. Same thing, if you go to 1000 and 1001, difference is only 1 Dalton, but the resolution required is m by delta m, right? Now 1000 by 1. You need 1000 resolution to differentiate these two, okay, to separate these two. If you go to 10,000 and 10,001, difference is 1 Dalton only, but you require 10,000 resolution, okay. So as the mass value increases, the difference, same difference, you require high resolution to be separated. So this is the point. So I'll tell you with the importance of high resolution with one example. Okay, it's a standard example. Normally, everybody will give you. Assume there are 3 molecules: carbon monoxide, nitrogen, and ethylene. The nominal mass value is 28 for all. Okay, if you have a low resolution instrument, you cannot separate these 3 molecules. Okay, then you have a high resolution instrument. Okay, for example, you have a 2300 resolution instrument and recorded the spectrum of this mixture. Okay, if you have 2300, you can separate ethylene but not carbon monoxide and nitrogen. Okay, you can see the resolution is not sufficient to separate these two. But if you apply 5000 resolution, now all the 3 are separated. You have a carbon monoxide, you have a nitrogen, you have a ethylene separate. Like this, if you have mixture of different molecules having same nominal mass, if you have a high resolution instrument, because the decimal point value is different, you can get separated and you can detect easily what is that. Okay, so that is the important of high resolution technique. So I'll give one example here, where this is a biological sample which was taken mass spectrum. Without chromatography, it was taken the spectrum. Okay? That means all the molecules will come together. So here one example you can see, there is a glucose in the sample, and there is a spermine which is a metabolite whose nominal mass is 202 only, molecular weight M plus S is 203. Okay, but if you see the accurate mass, okay, the one is the 20 exact mass, if you see 203.0532 for glucose sodium adduct. If it is a spermine M plus H, it is 0.2236. Okay, so if you apply high resolution, then you get 2 peaks. In the normal low resolution, you get the single peak. Okay? So that's how, by using high resolution, even though there are 2 molecules are there, same nominal mass, but with different accurate mass, you have separated this one. So this is the advantage of high resolution. But thing is, what kind of instrument you are using. If you are using magnetic sector instrument, you can get 80,000 resolution with 10% value. Time of flight 40,000, but recently one reflector top is there. I think that can come 80,000. But RB trap is a little less expensive. But now I think it has gone to 2,40,000 resolution. FTMS is always very high, 5 lakhs resolution, but only problem would be maintenance. So I don't think any FTMS, but we don't bother about FTMS. But a normal is orbit trap and time of flight. These are the high resolution instrument nowadays people are using. Okay. So you can see I just kept the high resolution instruments. One is the magnetic sector, very big. Still people are using. I don't say it is not completely abandoned. This is used now for dioxin samples. Very specific application. QTOF is a common high resolution MSMS instrument. Nowadays we have a Q exactive Orbitrap instruments where you can also do high resolution MSMS. The only difference is Orbitrap will have low mass value about 2000, 3000, whereas others they don't have mass limit. This is FTMS I was telling. This is a ion cyclotron resonance, but it is very specific use. Same thing, it is tabulated here. What kind of instrument, what is the resolution, what is the ppm error you get. Nowadays, all the high resolution instruments, they are giving 1 ppm, less than 1 ppm error. Of course, the accepted value is 5 ppm. So once you have the elemental composition by HRMS, you can get double bond equivalents where number of double bonds, number of rings you can get. As a chemist, you know what is DBE. This will give some such information. Okay, I just gave some example. I can skip this one. The next one is important one is the isotopes. Okay, as I said, all analyzers low molecular weight below 2000, it will nicely separate isotopes. The first one is not, if it comes to the organic molecules, the first one is the C12 peak, and M+1 peak is because of the C13 isotope, and M+2 normally, it can be from oxygen, it can be from silica. I have given here what is the M+1 contributions and M+2 contributions. But major M+2 contributions are from silica, sulfur, chlorine, bromine. Okay, these are the major contributions. Otherwise, it will be a small peak. Okay, if you have a chlorine, it will give you 33%. 1 chlorine, 2 chlorine, 66%. Like that it will go. And if you have a bromine, very clearly the M+2 will be 1:1. So like that, the isotopes are very important in the structural elucidation. The first, the M+1 peak, if you divide it by 1.1, you get a number of carbon atoms straight away. And M+2 will give you some heteroatom information. For example, let us say the isotope C13 isotope. As the number of carbons are increased, for example, if you have a molecule with carbon 50, then your M+1 will be 55%. So by looking at M+1 peak 55%, you can immediately say there are at least 50 carbons in the molecule. But if the number of carbons are increased to 100, your C13 will go up. It will be higher than C12 peak. So especially when you have a protein, then of course you get average mass, and also C13 peak will be coming up. Okay, that you should remember. So this is simple calculations. I will not go through, but if you have C13 contribution 1.1%, then you can get number of carbon atoms. Okay. So that is the one thing. The other thing you apply is the nitrogen group, because whenever you want to characterize your molecule, you have to get first molecular mass. Okay. In order to apply nitrogen group, you have to get molecular mass. Okay. For example, if you LCMS, you are getting M+H. Then proton you have to remove to get molecular mass. Otherwise, if you are getting a sodium adduct, M+ sodium, then 23 you remove, you get molecular mass. Okay, the nitrogen rule is applied only for small molecules less than 600, and you have to get your molecular mass to get nitrogen rule applied. Okay, if your molecular mass is even number, then that means you have even number of nitrogens. Remember, 0 also even number. Okay. But if your molecular mass is odd mass, then there are odd number of nitrogens. Odd means 1 nitrogen, 3 nitrogen, 5 nitrogen, like that. Okay. So this is the nitrogen rule. Because why this very special about nitrogen is the atomic weight and valency. In all other cases, if atomic weight is even, valency also even. But especially when it comes to the nitrogen, the atomic weight is even, valency is odd. So because of even number of nitrogens, odd mass value, odd number of nitrogens. So it is applicable to only molecular mass. Of course, it also applicable to fragmentation, but you have to be careful, when nitrogen is there in the fragment or not. But it is very complex nitrogen rule when it comes to the fragmentation pattern. Roughly, of course, you can apply to the main mass value, molecular mass value. Okay. The other characterization point of view, if you get the mass value, you can apply rule of 13. Okay. Rule of 13 is very simple. Okay. You get the molecular weight divided by 13. Okay. When you divide by 13, it may not be integer value. Okay. So it will be divisible plus you get remaining value. Okay, I'll give an example later. So if it is divisible, that is n value. The remainder will be r value. Okay, n plus r divided by 13. Okay, the formula will be Cn H2n plus r. Okay, with example you can get easily. Okay, then if you get a n value r value, you can get double bond equivalent by this formula, n minus r plus 2 divided by 2. Let us see one example. You can see. Okay. Now you have molecular weight. Okay, example is here. Your molecular weight is 94. So if you really see 94 divided by 13, how much it will come? 94 divided by 13. It is 7.2 is coming. Okay. That means 7 is the actual value. Remainder, there is some remainder. Okay. A remainder 94 into 7 is 94 divided by 7 is so 13 into 7 will be your 91. Whereas 3 is remaining, and 3 by 13. Okay, you have a n value now, r value. Okay, n equal to 7, r equal to 3. But now molecular weight 94. So that means the base formula what you have is C7H10. Okay, then from this you can get double bond equivalence. 7 minus 3 plus 2 whole divided by 2. That means it has the double bond equivalence 3. Okay. So by using this rule 13 value, you have now a basic formula and you also have double bond equivalence. And if you want to replace with a functional group, the tabular form I have given here. If you want to replace with nitrogen, for example, you remove CH2 from this and add nitrogen. Like this, there is a subtract element. You can get some information. Okay. So this is what about the mass spectrometry and some characterization point of view, nitrogen rule, high resolution, like that. But if you want some more, I can tell you, but I don't know what is the time now. If you want close, then I can stop it here. Dr. Anush, Dr. Lakshmi, madam.
Yes, sir.
Yes.
Yeah, if somebody wants, then I can tell rearrangement and those things. Otherwise, we can stop it here.
Okay.
If you have 5 minutes, then I can.
Yes, sir.
Add some
5
more
minutes is okay, sir. No problem, sir. Yes, sir.
Okay, another 5 minutes. Okay,
Yeah,
I'll
yes, sir.
quickly go through. If you see isotopic patterns, I already explained you. There are certain rearrangements in mass spectrometry. Okay, so if you have two functional groups in very close proximity, we say ortho. Okay, then you can get neutral loss, very specific neutral loss because of interaction between two molecules. If the same thing in meta and para, you cannot see this kind of neutral losses. Same thing will apply for HP LCMS. It is in GCMS, but similar neutral loss you can see in LCMS also, soft ionization also. So this is ortho effect. The other one is alpha cleavage, beta cleavage. Normally in EA fragmentation you can see, but even M+H ion also you can see alpha cleavages. This is some kind of fragmentation pattern. But if you, there are some specific rearrangements in mass spectrometry. Okay, one is the retro-Diels-Alder reaction. Okay, where if you have a cyclohexene type of structure in your molecule, then it will rearrange with to give diene and dienophile. Okay, that means one fragment with two double bonds, the other fragment with one double bond. Okay, you can have a charge either this side or that side depending on the stability of your molecule. But actual Diels-Alder reaction is diene dienophile will give cyclohexene. But in mass spectrometry, it is reverse. Cyclohexene will give diene and dienophile. That's why it is called retro-Diels-Alder reaction. It can happen in M+H ion also. Okay, this is very important for natural products. There are some structural characterization. The other one is the loss of phenol from loss of carbon monoxide from phenol. Very specific loss. It can happen in EA and it also can happen in ESI. Other thing is the McLeod rearrangement. Everybody knows if you have a unsaturation and if you have a gamma hydrogen, this gamma hydrogen will shift to the oxygen and it will rearrange. Okay, you get either this is protonated or this is charge returns depends on the stability. But after rearrangement, you get two fragments. One is neutral, other is ion. Okay, McLeod rearrangement is very famous in mass spectrometry. People think only in EA it will happen, but McLeod rearrangement can also happen in elsewhere. Wherever this gamma hydrogen is there, even M+H ion also you can see McLeod rearrangement. So this is very important. So thank you. He is a McLeod. I met him. I am happy to see him because in routine characterization, no, I use McLeod rearrangement and retro-Diels-Alder rearrangement. These are the two important rearrangement in mass spectrometry. So thank you all. In my lecture now, I started with basics, given what is the source analyzer detector, and I touched high resolution, important of exact mass and accurate mass what you measure. And MSMS I have not touched, but because of high resolution instrument, you can do even fragment ions also, you can get elemental composition. So thank you.
Thank you, sir, for this very interesting lecture where you discussed, as mentioned by you, the fundamentals and the theory behind it and the major parts of a spectrometer, MR spectrometer, spectroscope, etc. So thank you very much. And now the session is open for discussion. The participants, if you have any questions, please ask it. You may either type it in the chat box or question answer box, or you may also raise your hand. Control room, do you see any questions in the chat box or the question answer box?
Sir,
no,
I have a question. So in that last slides, you are talking about carbon monoxide loss. So that is
Yeah,
a very standard phenomena, that ring contraction.
yes, yes. It is a standard phenomena with phenols. Actually, phenols are specifically looked for this carbon monoxide loss. Mostly in GCMS, but in certain implicit chain also, you can see. I have seen many examples. You have to be careful, no? You may be thinking that there is a carbonyl group in the substrate, but if you have a phenol, you should be careful.
Okay. But it is only very specific to this kind of aromatic phenols, or other six-membered ring systems also, like
No,
not
only for
aromatic.
aromatic phenol. Aromatic phenol.
Yes, sir, thanks.
And other one actually I have not covered. If you have aniline,
Sir.
that will lose SCN, cyanide, SCN. Like
Okay.
carbon monoxide, no? Aniline will give you SCN loss, 27.
But that ring contraction will occur there also.
Yeah, there also ring contraction. Because aniline is pH NH2, no?
Okay.
Phenyl group connected to NH2, right?
Yes, sir.
Now, like carbon monoxide in phenol, here CN will go.
Okay.
That also very specific loss.
Okay, sir.
Any other questions from the participants?
So I can share the PDF file if they want, students want.
Okay. We are also recording the session and it is available to all the participants actually. So,
Okay, okay. And if it recorded, then no issue. They can.
yeah, it is all sessions are getting recorded. So yeah.
Okay.
Okay. So if there are no more questions, let's thank the speaker for this very informative lecture on mass spectrometry, which covered almost all the major aspects of the instrumentation and the fundamental theory of the technique. So thank you once again, sir, for joining us today. Thank you very much.
Thank you, madam. Thank you, Anuj and Dr. Lakshmi.
Yeah, thank you, sir. Thank
for
you very much
giving
for accepting the invitation. Thank you, sir. The committee room, you can.
Thank you very much, sir, for the very detailed explanation on mass spectrometry. And dear participants, now it is time for a break. Please have your lunch and join back to the platform at sharp 1:45. Thank you.
I think it's a good session. Exactly stopped at 1.
Yes, sir.
Okay.
Sir, I
Yes,
have
sir.
a, yeah, see, where is Vairamani, sir, these days?
Yeah, he is in SRM now. Yeah, like
Okay.
SRM University, Chennai.
Okay. So he retired from there and moved to Chennai.
Yeah. I think I already completed more than 10 years now. He retired.
Okay. Yeah.
So now I am
Thank you.
taking care of the mass center.
Okay.
After Dr. Vairamani, Dr. Srinivas was there.
Uh-huh.
For 3 years back, he retired.
Okay.
So I am taking care of and looking.
Mm-hmm. Okay. Thank you, sir.
Hello, Arjuna.
Yeah. Anu,
Yeah.
Dr. Anuj?
Yes. It's audible.
Okay,
No issue,
nice.
but you need you want to check the presentations?
Yes, just I will
One
check it
second.
once.
One second.
Dr. Anuj,
Yeah, just to hold for 1 second.
you are giving permission, huh?
Yeah. Actually, I've given to the students. I'm calling them. They have to. 1 second. I'll just leave and join for 1 second.
Yeah, okay.
Yeah. You got it. Okay.
Yeah, 1 minute.
Yeah.
Now is it okay?
Yeah. It's coming. Yeah. Okay.
Shall I put it like this?
Yeah, it's fine. So just put it on the presentation mode.
Yeah.
It's fine. Good. No issues.
Shall I keep or shall I close?
You can minimize it because that other slides will come.
Shall I close?
You say stop sharing.
Yeah, 1 minute. Stop sharing. Okay, bye.
Okay.
Okay, Dr. Anuj.
Yeah. I'll under 15 minutes I'll join. Okay.
Sure.
Hello?
Sir, hello, sir.
Yeah. Yeah, Vikrant. Yes, sir. I think Nagarjuna is also there. Our committee team, we can start.
Yeah,
I think Nagarjuna will start.
Yeah. No,
Because
actually, our conference secretariat is there. So,
Yeah,
sure.
Dear all, welcome back. Moving on to the next session, we are going to have a talk on NMR data analysis by Dr. K. Nagarjuna and Dr. Vikranti from Advanced Center of Research in High Energy Materials, University of Hyderabad. I cordially invite Dr. Anujay Varghese, Assistant Professor, Department of Chemistry, NIT Calicut, to share the session. Over to you, sir.
Yeah. Thank you, Anushree. So, Dr. Nagarjuna is already has been already introduced to the participants. He has already handled one of the session on the second day, and Dr. Nagarjuna did his MSc from IIT Delhi and completed PhD from the University of Hyderabad, and currently working as a postdoctoral fellow at Advanced Center of Research in High Energy Materials at University of Hyderabad. And Dr. Vikrant completed his MSc and as well as PhD from University of Hyderabad, and currently working as a postdoctoral fellow at Advanced Center of Research in High Energy Materials, Akraham, University of Hyderabad. So I welcome both the speakers, Dr. Nagarjuna and as well as Dr. Vikrant, to take over the session. Yeah, you can start.
Dr. Anuj.
Yeah, you're audible. Yes.
Yeah, okay. Thank you once again, Dr. Lakshmi C. and Professor and Anuj Varghese, giving this opportunity. First, I'm sharing. Dr. Anuj.
Yeah, it's visible. It's visible.
Yeah. Once again, good evening to all. We have already seen last time the importance of proton NMR, and then some analysis of some few compounds and data analysis. Today we are going to look at data analysis of some C13 NMR and F19 NMR in some case studies. But F19 NMR spectra I did not record, but mostly I will discuss on C13. And for example, like combination of carbon fluorine compounds, how it split in the C13, I will discuss. It's very interesting one. Now I start with importance of C13 NMR. We know proton NMR is we know that the natural abundance is almost compared to C13 NMR, it is more the abundance, and also the gyromagnetic ratio of C13 NMR is very low because it's an intrinsic property of the nuclei, and that's what usually it required more scans. For example, it's like 1/4. Suppose if I am using a 300 megahertz for proton NMR frequency, but for C13 it will be like a 75 megahertz. For example, 600 means it is like a 151 megahertz. That means very low frequency. That's what we knew. For C13 we used to give a more number of scans, because generally carbon detects only C12 isotope carbon, whose natural abundance is only 1.1, but the main carbon is C12, but unfortunately it is not NMR active. Whereas C13 NMR signals does not split like proton, but the it will give number of signals equal to the number of lines in the C13 NMR spectrum. And mainly there is a lot difference between proton and C13. Proton NMR is to study the number of non-equivalent protons present in unknown compound, whereas C13 NMR is to determine non-equivalent carbons and identify the types of carbon atoms also. And C13 signals are spread over a wide range than the proton signals. Why? Because when suppose if we are taking a carbon attached to any electronegative atom, even though hydrogens will affect less when compared to carbon. Carbon affects more electronegative effect on that attached carbon. That's what we used to get all the downfield in the chemical shift ppm values. And another important thing is that by proton NMR, one useful thing is that we can identify the it's very sensitive, because that's what sometimes we used to get very impurities also in proton NMR. But whereas C30, you will get only the particular the major compound peaks only you will get. So that's what C13 is a very important tool to confirm the some particular products. So I will discuss some of the few things. So typical chemical C13 chemical shift regions are major. These are the classes. For example, ketones and aldehydes. There it will give around 220 to 200, and carboxylic acid esters, and then methanes and aromatic olefinic nitriles, aromatic, and as if you see the ranges from 0 to 220. This is the typical range for organic compounds. So just I want to discuss why it is so important when compared to the proton NMR, because proton NMR only it gives number of protons, and sometimes it is very difficult to confirm your reaction, your product by only proton NMR. For example, if you take this reaction, if you are taking this kind of without hydrogen containing this kind of trichloro triazine compound, when you couple with simple pyrrozole, see in proton NMR we will get only 3 peaks. So sometimes we do not know whether this reaction occurred or not. Sometimes we don't know these chlorines are converted to not form, only pyrrozole is there, then only you'll get same number of 3 peaks. So sometimes we may first whenever we see NMR, we feel that maybe compound was formed, but sometimes and again after some analysis we will come to know that it was only pyrrozole. But C13, if you, because once it is coupled, these carbons will come one region, and these 3 carbons will come. Definitely 4 very strong 4 peaks you will get. So that's the way C13 will resolve your product conformations in some of the cases. And also, so for example, and some in some cases very complicated cases also, C13 will give very it's a technique to identify your expected product. For example, I taken this compound, I did reduction, and then I know by somehow initially I will do some TLC studies, but yet I need to confirm accurately. So for example, after proton, so there is a proton, you will get lot of peaks. Sometimes it is very difficult to assign number of protons, because you see this CH2 and here this CH2 and again the benzyl, there is a CH2, all are like sometimes it becomes a mixed places. It is very difficult to exactly isolate the number of protons. But if you see C13, if you go for C13 via this keto compound, particularly these two will come around 158 ppm, and this one will come 178. After reduction, we know that these were converted to CH2 saturated. Definitely this value it will come around 150, 140 ppm, and this is 137. So that one, it will see that will be a clue to that my reaction was exactly formed my expected product. So that is the way C13 very useful. And another thing, if you see this one also, suppose if you are taking simple benzonitrile and converted to by using zinc chloride and surajite, this kind of tetrazole, and generally we all know NH is very difficult to sometimes to get in NMR. But so by based on proton NMR, we cannot say that 100% my product has formed. But if you see when you go to the C13 NMRs, here the nitrile C triple bond, the carbon it shows 180 ppm, but whereas after forming the tetrazole, in this particular carbon, it shows the value 155 to 150. That is the one of the key peak for to say that my product was formed, because you cannot confirm all the structure as XRD, because sometimes XRD we cannot get all the for to grow the crystal structures. But somehow this C13 will give you to resolve your products conformation. See, because now if you see, you can do a number of derivatives. For example, if you put in benzene, maybe you can synthesize some 100 or 200 compounds, and in every C13 you need to get this 158 to 150 is a key peak. If you are missing this peak, you can assume that maybe the CN was not converted. So these are the typical examples I took. It's a very large we can study on C13, but I typical with my experience, I took some typical examples. So such a way it will resolve your product conformation rather than the proton. Only it depends on only C13 NMR. So now today we will discuss some compounds. Like already last time I discussed this paramethoxy benzaldehyde, and this is a toluene, and this is a glycol ester, and also some fluorine containing compounds I collected, and then we'll see that how it will show the C13 NMR and what are the differences. For example, if you see here, C13, every carbon, for example, toluene, if you consider, this methyl shows low chemical shift. It's like upfield. But if you see the carbon, methyl attached carbon is around 138, and these two are ortho is 128 and meta is 128 and 125. So the reason is that the environment of the carbon is it will split into different places. Whereas this carbon is attached to the three hydrogens, it's like a tetrahedral carbon. This is a tetrahedral carbon. So that's what it is formed in a low ppm in chemical shape. And whereas this carbon attached this one, it is a very weakly affected by the strength of the magnet. That's what it is 138.4. And whereas these two, and of course these two are almost all equal, but still there is a little difference because the methyl group on effect on this one is more and this one is less, and whereas para is entirely again different. So like that, in C13 we can identify that nuclei of the different nuclei in the carbon. So now these are the typical examples I taken. And another thing, if you see the fluorine cases, most of the, if you take any fluorine, you will get particularly because we know fluorine also NMR active of 19, and when it is attached to the aromatic system, and if, you know that this carbon also see that inactive, so it will more all the fluorine compounds you will get doublet peaks. So this fluorine will influence more on here and less here and less here and less here, and sometimes even it affect here also, but whereas it will not affect, but in case symmetrical molecules it will affect in the long range also. Whereas CF3 also, this if you take this carbon, it was surrounded by 3 fluorine atoms, and C13, that's what this fluorine, and according to the n+1 rule, so this carbon, particularly this whatever wherever I pointing here my cursor, you will get a quadrate peak, this carbon. And this also, these 3 fluorines will affect here also, and even affect here also and here also. So that's what majorly the more effect on this attached carbon here. That's what this carbon, you can see nice quadrate. So that even that that quadrate, the H value, it will come around any CF3 compound, the H value of the directly attached 3 fluorine carbon, it will come around 260 to 270 hedge. That is it is a very important. It has to match that around 260 to 270 hedge of the coupling constant. And next that next attached this carbon surrounded other three fluorine atoms, it will affect around that small quadrate will get, that around will get some 30 to 40 hedge is the coupling constant. And again these fluorines will affect here and here also, it very less because it's very far. So it will around, it will these carbons also sometimes will give quadrate. It's a very small quadrate. We can see in C13 NMR very nicely. So that coupling constant around some 3 to 5 megahertz. So like that you can nicely see the coupling. Because why? Because nowadays a lot of CF3 introducing compounds are coming. So when you are taking some reagent, you are conducting reaction. The confirmation that your CF3 was attached on that is by checking the cystatin NMR. So that is one of the very important tool. And for example, if you are taking this last compound, we put here is in between we put an oxygen. So definitely, if you see here, these fluorine atoms only affect on this carbon. It will not affect here and here because there is in between on oxygen. So like
Yeah,
that we
but
can.
Nagarjuna, can you just take
Yeah.
the pointer
Yeah,
instead of mouse cursor?
1 minute. Now it's okay.
No,
can
just right click on that one.
1 minute.
Just right click. Yeah,
Yeah,
you
yeah.
take pointer options.
Okay, okay,
Yeah, take laser pointer. Laser pointer you take.
okay.
Oh, okay, that is also fine.
So here this carbon around surrounded by 3 fluorines, it will affect here only on more, but it's very difficult to affect here and here because in between there is oxygen. So like that it will be useful. Now I will show the actual how the spectrums by using the Delta software. Just a minute. I will stop the sharing for now. Yeah. So now this is the 12-in. Dr. Anuj Varghese, it is visible. Dr. Anuj.
Yes, sir, it's visible.
Yeah, okay, okay. So this is the C13 of 12-in, and then we can in GeoL directly we can, because C13s are a very wide range of ppm values, so we can select this one. The corresponding it will come the peaks, and then you can give. And this is the DMSO solvent, whereas in proton NMR we will get around 2.5 is the corresponding the trace amount of CH3 S11 CH3, whereas in C13 we will get in around 40. So then you can see here, I told you already, in toluene the total this methyl carbon, tetraethyl methyl carbon, that carbon will come around 21.5, and then the CH3 attached carbon peak it will come around 137, and then this ortho and this meta carbons, because they are coming equivalent carbons, that's what it is. 2p intensity was double, and this 125 is the para carbon. So this is about the 12-in. And this is a simple hydroxy, So last time I already discussed you when we collected an in DMS or D6, the OH corresponding 1,4-dihydroxy engine. So the OH carbons, it will come attached carbons because more alternative atoms, it has come as around 158, and the remaining 4 carbons of the 2 ortho and meta carbons all will come as in a one intensity because it's a symmetrical molecule. So that's what we will get only 2 peaks, and then This is already, you can see this 4-methoxy benzoylate. So that C double bond O is a 4-methoxy benzoylate, and we can see that C double bond O benzoyl CH4 carbon, it will come around 198, and then remaining carbons, we can see the methoxy, it will come, because methoxy carbon will come little downfield than the methyl, because it was surrounded by the oxygen, whereas in methyl, see, directly attached is phenyl to methyl, there is no much electronegative effect. That's what it comes around 21 or 23. But whereas most of the methoxy groups, the CH3 it will come around 50 to 65 range. So these are the some typical nucleus you can see, and then this is glycoviryl diethyl ester. I already told that because two carbonyls I have, so one is 167 and 160. So and another that one aliphatic carbon is there, that is 78, and then this is ester group is there, CH, and this is a CH2 and this is CH3. So like that we can see, this is I told already, if you do some reduction on these carbonyls, definitely these will when it converts to C double bond or to CH2 saturated compound, then we can see the peaks shifted to here. And now I will go to the fluorine compounds. It's a very quite interesting. So this is simple para-nitro, parafluoronitrobenzene. You can see I got here doublet, here doublet, here doublet, here is the doublet. So now the fluorine attached carbon, it was doublet here, this is the one. So it, because it was directly attached to that carbon, that was its effect more. So this chemical shift, this it's a 2 doublet. If you see the signal and then that edge value, it will come around 160 to 170, and this remaining here one and here one and here one, total 3 is there. So I told you already, it will affecting the direct attached carbon, and then next alpha carbon and beta carbon also it will affect. And this side also we have a 2 alpha carbons are there and 2 beta carbons are there. That's what it affect here, 2 these doublets, and even it affecting the meta also. So that's what here this we can see, this is 118546 minus 118.412, and if you calculate the corresponding, see that in 151 if you multiply, it will come around some 5 to 6 hedge. So like that we can see. And then again in NMR, if you don't want the fluorine coupled, in such case you can do C13 NMR with the fluorine decoupled also. You can study that also. That already I also, that one I will show you. Fluorine decoupled. I think I did not put here 1 minute, but I have a spectrum here. If you see, this is a fluorine decoupled. When I did fluorine decoupled, I got all singlet. The same parafluoronitrobenzene C13 with the proton decoupled. Of course, obviously we are doing proton decoupled only, but with again I did F19 decoupled. It shows all are singlets. So this is one thing we can observe in this one, and then I will discuss you now about the CF3 group. Now I already showed you this one. If you see this CF3 group, this is one 3-chloronitro benzotrifluoro compound. First you can assign all the peaks by directly, then give. And now I told you directly attached CF3. You can see nicely directly CF3 attached carbon. So this is the quadrant. 1, 2, 3, 4. This is the quadrant. Then you can see difference 125.220 minus 123.410. If you do these two difference, you will get almost the equivalent around 260 to 270 H. And then how you calculate means you just difference and then multiply with the frequency, that is 151 megahertz. And then if you take this 123 and 121, these two also it will come same, and these two also it will come same. This is the directly attached carbon. That's what it shows, a very high megahertz value, that coupling constant. And this is a next attached, like the other carbon, which I told you already, it will come some 35 to 40. You can see this one, and this 133, and you can see nicely this quadrant. So most of the CF3 compounds, you can see this kind of splitting. So whereas because this another functional groups also there, that's why it is not affected on other carbons. It was only affected on the directly attached fluorine attached carbon, and another beta carbon also it is attached. And another thing I will show you, this is amino. This also testing. You can going to try for this one. Chromo CF3. Yeah, you can also see this one. You can see here the quadrant. You see this small one here. This one, this one, this one, this one. So like that, it will be hide also. We need to be very careful by selecting, because that only we can confirm by the differences of the that hedge value. And this is another small quadrant. So like that, we can nicely we can see in every sector number. And lastly, I want to discuss this trifluoro OCF31. So I already told, since we put OCF3, so that's what it will come only one quadrant. It will not come other, because it will be, since oxygen atom in between is there, it will not affect other carbons. So this one also we can clearly we can see this 123 and this 121, 119. So like that we can identify. So this is the one I want to discuss, but still I would have collected the fluorine NMR data. But, the stipulated time I covered this part only. Yeah, so I want to thank you for giving this opportunity. Over to you, Dr. Anuj Varghese. Dr. Lakshmi. Dr. Anuj Varghese.
Yes, sir.
Yeah, my session is over, madam.
Okay. Thank you.
So just I'm stopping my sharing. Thank you so much for giving this opportunity, and thank you all.
Hello. Yeah. You have completed?
Yeah. Yes. I completed my part.
Yeah. So Vikrant is ready. You can take over.
Yes, sir, I am ready, but I need access for sharing.
Yeah, you give the permission for sharing. No, he's already in the panelist. You give only the presentation. Yeah, you have the permission now. I can go for it.
I think my slides are visible.
Yeah, you can just put it in the presentation mode.
Thank you, Anu sir, for the introduction, and a very good morning to all. So, and I'm thanking for the organizers for the opportunity to present about some part of my PhD work that I have carried regarding 15-nanometer NMR spectroscopy studies, because it is a very important tool for our studies in the molecules of high nitrogen containing compounds. So this is an excellent initiation of this workshop for newly joined PhD students. I hope so they will learn some of the techniques and some theoretical background about this NMR analysis. And I think they must utilize these different types of techniques in their studies during their PhD. Today, I am here to present about the importance of 15-nitrogen NMR spectroscopy and analysis of 15-nitrogen spectra in structural analysis. So until now, we heard about proton and 13C NMR and some protein-based NMRs and biomolecules. So now I'm going to present about the 15-nitrogen NMR regarding the highly nitrogen-containing compounds. How do we analyze the nitrogen-containing compounds by NMR spectroscopy? My outline of presentation contains introduction, which tells about the importance and the differentiation between the 14-nitrogen and 15-nitrogen NMR spectroscopy. And second, methods for measuring 15-nitrogen NMR spectra, which gives brief technical details regarding the spectrometer. And then technical details, that is referencing of all the chemical shapes for different types of nitrogens. And also the theoretical analysis which can be done for a 15-nitrogen NMR spectroscopy, and we can compare this theoretical analysis with the experimental analysis part. And I'll show you some of the experimental results that I have got during this 15-nitrogen NMR studies. As I told you, adding introduction, this 15-nitrogen NMR spectroscopy is a powerful tool for the structural analysis of nitrogen-containing compounds, especially. So nitrogen is an important element in organometallic and inorganic chemistry as well as in biochemistry. It combines with practically all elements of the periodic table. And nitrogen has two NMR active nuclei, that is 14-nitrogen and 15-nitrogen. So nitrogen 15 is frequently used in NMR because unlike the more abundant nitrogen 14 that has an integer nuclear spin, that is 1, and thus a quadruple moment. But 15-nitrogen NMR, 15-nitrogen has a fractional nuclear spin, that is 1.5, which offers advantages over NMR like narrower line width. So that we can see how we will get 15-nitrogen NMR spectra and 14-nitrogen NMR spectra. So the 15-nitrogen NMR parameters are routinely measured in solutions at the natural abundance level of the 15-nitrogen isotope, that is 0.365, is two-dimensional NMR techniques. Here we can see the difference between the 14-nitrogen and 15-nitrogen spectra of urea especially. And we have seen 14 signals are usually significantly broadened by the quadrupolar interactions, sometimes to the extent that they are unobservable on a high resolution NMR spectrometer. Here you can see 14-nitrogen NMR, it is a broad peak. So because of its quadrupolar interactions. The 15-nitrogen NMR yields sharp lines, but it is very insensitive and are comparable with those of proton and 13C NMR spectra. Therefore, 15-nitrogen NMR nucleus is preferred to that of quadruple 14-nitrogen. This table represents 15-nitrogen nucleus basic properties of selected nuclei important for the organic NMR spectroscopies. So as we can see, isotope proton NMR, its natural abundance is 99.99%, and the natural abundance of 15-nitrogen is of 0.365. And its sensitivity, relative sensitivity is only 0.022 when compared with the 14-nitrogen, that is 5.69. So there are several methods for measuring 15 nitrogen NMR spectra. As 15 nitrogen NMR spectroscopy has inherently low sensitivity, and the energy difference between the nuclear spin states are of the order of 10 to the power of -5 of the thermal energy at room temperature. Therefore, enhancement of the NMR signal is one of the major goals in NMR spectroscopy. So how we will achieve this enhancement of NMR 15 nitrogen NMR signals in NMR studies? So as given by the equation, the sensitivity, that is signal to noise ratio of one-dimensional experiment involving nuclei with a spin half, can be expressed as follows. So here we can see the signal to noise ratio is directly proportional to all the terms in the equation. So where n represents the number of molecules observed in the sample volume, and a represents the abundance of the NMR active spins involved in the experiment, and t represents temperature, and gamma excitation and gamma detection represents the magneto-gyric ratios of the excited and the detected spins respectively, and B0 is the static magnetic field, and T2 is the effective transverse relaxation time, and NS is the total number of accumulated scans. So the signal noise ratio is directly proportional to all of these terms. So how we can enhance the signal to noise ratio in 15 nitrogen NMR according to this equation. So in the equation, if you can see, if we increase the number of molecules n by increasing the concentration of the sample at a fixed volume, or increase the observed sample volume at fixed concentration, so that signal to noise ratio will be increased, or increase the abundance of the NMR active nuclei A by enrichment of nitrogen, or we can increase the static magnetic field, that is recording at higher magnetic strength, or increase the NS, that is the total number of scans, accumulated scans, either by increasing the time for accumulating the scans. So we need more number of scans for getting the sharp peaks and a signal to noise ratio for a 15 nitrogen NMR. So these are the different methods so that we can increase that signal to noise ratio in 15 nitrogen NMR. So there are two principally different methods can be used for the measurement of this 15 nitrogen NMR parameters. One, the direct detection of an 15 nitrogen NMR spectrum is most commonly used by chemical community, and this approach suffers from very low sensitivity because of its low abundance. So gamma excitation is equal to detection, that is equal to nitrogen, we can get that peak. And the second method is experiments based on the excitation and the detection, that is called inverse or indirect detection, using that proton represents the most powerful approach for the determination of the parameters of 15 nitrogen NMR. As here we can see, the gamma excitation and detection is equal to proton. So proton coupled NMR by indirect detection can be used for the 15 nitrogen NMR parameters. One is direct detection and other one is proton coupled nitrogen. So what is direct detection method? It is the most common technique, as I said, it can be used by all of the chemists for different types of nitrogen atoms. But unfortunately, it suffers from the low natural abundance and low and negative magneto-gyric ratio, and also low relative sensitivity, and often also from long relaxation times. So what are the requirements to increase that signal to noise ratio required? So as we can see, long accumulation of times operating at high magnetic field are required to get the better spectra. And also nitrogen 15 enrichment can be used to overcome the problem of low sensitivity in 15 nitrogen NMR spectroscopy because of its abundance of 0.37% only. And the relatively long 15 nitrogen relaxation times typical for the nitrogen atoms in small molecules can be reduced by using relaxation agents like chromium triacetyl acetonate. It is the compound most frequently used for this type of purpose. Several direct detected experiments differing in their application of proton decoupling can be used for this purpose. When no decoupling is applied during the pulse sequence, proton coupled nitrogen spectra can be observed. So that removing all of the proton 15 nitrogen couplings in a proton decoupled experiment concentrates only on the nitrogen resonance into a single line, thereby increasing the signal intensity significantly and simplifying the spectra. As you can see in the figure, the 15 NMR recorded for this compound number 1 by using 500 milligrams of compound in deuterated chloroform 2.5 ml by using 10 mm NMR tube, and data were acquired on a broker of 4,600 scans were accumulated, and the relaxation delay T2 was 7 seconds. So here we can see nice narrow lines. So for 15 nitrogen NMR for compound number 1. This spectra can be obtained by the decoupling of proton, decoupled proton experiment. So what is inverse gradient detection method? In inverse detector experiments, the proton nucleus is used for both excitation and detection of the NMR signal. So this approach introduces gamma H as the gamma excitation, gamma detection terms in equation of signal to noise ratio, and thus increases the sensitivity by the factors. So at present, the most popular technique for multiple bond or long range coupling heteronuclear chemical shift correlation is HMBC, as can be used. So HMBC, as you heard about HMBC experiments, it is a 2D NMR, and we can get the long range coupling chemical shift values in this experiment. So as we can see, inverse gradient detection by inverse gradient detection method, the spectra looks like this. So this is HMBC analysis of one of the nitro derivative of pyrrole with an eliminated compound. As you can see here, we will find out the long range interactions between the proton and the nitrogen. So as you can see, this amine peak, it appears as 164.75.63 ppm, but the nitrogen of pyrrole, it appears as 164.27 ppm. And also we can see here third nitrogen of nitro group was missing because it doesn't have any interaction with this amine proton of this amine compound and also the CH CH of the pyrrole ring. So the nitrogen of this third nitro group is missing, and 5 and 4th nitrogens are having interactions with that CH of the pyrrole ring. So it appears in this spectra. As you can see, 5 and 4, they resonate at 344 and 347 ppm. So by this HMBC analysis, it is a quick analysis, and we can get this type of spectra within 2 hours. Because a normal 15 nitrogen NMR, it requires a direct detection method. It requires more than 5,000 scans for 100 mg sample, and it takes a lot of time, almost 1 day to get the spectra of 1D 15 nitrogen NMR. But by HMBC analysis, we can quickly predict the number of peaks that is having in interaction with the proton. So here in this spectra, clearly we will not get any nitrogen which is not having any interaction with the hydrogen. So this can be used for a quick analysis when we perform reaction and we are waiting for the direct detection of the 15 nitrogen spectra. So this table represents the comparison of theoretical sensitivities of NMR techniques based on different excitation and detection schemes. So in the direct observation, as we can see in the direct observation, the 15 nitrogen NMR, its relative sensitivity was 1.0 only. But in the proton detected experiments, it is of 306. So by proton coupling, we can get the sensitivity of this 15 of the peaks which are having interaction with only proton. As I told, until now we have learned about the importance and the methods used for 15 nitrogen NMR parameters to get the 15 nitrogen spectra. So after collection of 15 nitrogen NMR data, how do we assign different types of nitrogens that are present in the compound, like proton and 13C NMR? Unfortunately, there is no universally adopted standard and chemical shift scale for 15 nitrogen NMR spectroscopy. The two most frequently used 15 nitrogen standards are liquid ammonia and nitromethane as a referential standard. The highly shielded nitrogen resonance of ammonia gives an NMR chemical shift scale similar to those of proton 13C spectrums. Most of other nitrogen resonances in the molecule studied are less shielded than that of ammonia. Thus, they will resonate at higher positive values of the chemical shift. So we can get all the peaks in the downfield, that is with a higher positive values. Unfortunately, this standard is not easily prepared in routine practice, and its chemical shift depends upon the pH and also the temperature where we are recording the spectra. So because of its historical use and ease of preparation, organic chemists often use nitromethane as a primary standard in referencing this 15 nitrogen NMR spectrum. Unfortunately, the position of the nitromethane nitrogen resonance produces mostly negative but also sometimes positive values of chemical shift for typical organic compounds when it is used as a primary standard. So in this nitromethane standard referencing, we get either the values in positive region also in the negative region. So other less frequently used standards are ammonium chloride and ammonium nitrate and nitric acid. So here you can see the standard reference samples that we can use for 15 nitrogen NMR chemical shifts. So this is the interrelation between the 15 nitrogen chemical shifts of several substances used as the 15 nitrogen chemical shift standards for a field at 25 degrees. As we can see, most of the 15 nitrogen NMR we will record at liquid ammonia, that is the reference peak will come at 0.0 ppm. So for referencing in nitromethane, we should reference that liquid ammonia peak to minus 381.7. Then we can convert to the whole spectra into the nitromethane standard reference. So in this way, we can interrelate other standard reference samples also. As we can see, nitromethane urea and urea to nitromethane, so ammonium nitrate and also ammonium chloride. So mostly we will report all the 15 nitrogen NMR spectra either in liquid ammonia or in nitromethane for publication purpose. And these can be accepted as a standard. So these are the characteristic range of 15 nitrogen chemical shifts in various types of compounds. As you can see, like proton 13C NMR, the range, whatever we will see the range of alkane protons or alkene protons or aromatic protons, how they appear in the range of ppm. So here also we can summarize all the nitrogens into the ppm range of different functionality. So as we can see, aromatic nitrogens will appear in the range of 340 to 385 ppm, and also nitro groups will appear in the range of 340 to 360 ppms, and tetrazoles, triazoles, they will come azoles, pyrrole like nitrogens. So any compound which heterocyclic compounds, the ring nitrogen will appear in the range of 120 to 150 ppm, and triazole, the nitrogens which are attached to the NH of the triazoles, they will come under 160 to 260 ppm. So in this way, we can identify and assign the different types of nitrogens of our compound in the NMR spectra. So these chemical shift values are completely referred to liquid ammonia only. So after recording the spectra at liquid ammonia, then we can reference to the nitromethane for publication. So these are the experimental techniques that we are going to use in the NMR spectrometer, and we'll get the data. So what is the need of computer assisted analysis in 15 nitrogen NMR parameters? So these ab initio computation of NMR spectroscopy parameters are used for last 2 decades for calculating these 15 nitrogen NMR parameters. The calculation of these parameters has become a routine tool used by a rapidly increasing number of non-specialists, because as I am a chemist, so I can assign the peaks regarding that shielding and de-shielding effects of the compounds which are present in the compound, the functional groups which are present in the compound. But for non-specialists such as spectroscopists and synthetic and natural products, they can analyze these 15 nitrogen NMR, experimental 15 nitrogen NMR through computer assisted analysis also. So the growing number of papers clearly shows that calculated NMR parameters can be used in a number of applications, such as assistance with assignment and prediction of unknown spectra, and also estimation of the separated shifts of coalescing nuclei and dependence of NMR parameters on intermolecular interactions and steric and solvent effects also. and also to study the principal components of the shielding tensor, etc. So a quantitative agreement between theoretically calculated NMR spectra of idealized model systems and routine experimental data obtained from a real system can hardly be expected. So whereas NMR parameters are calculated for zero temperature equilibrium geometries of free molecules in vacuum, but the experimental data are mostly commonly obtained using condensed phase samples, that is liquids or solids at ambient temperatures. So consequently, the experimental spectra are inherently affected by intermolecular interactions. So a large arsenal methods has been developed for the this type of calculations of NMR parameters theoretically. So it is generally accepted that the accurate prediction of these properties requires Gauss invariant procedures, that is having the same electric and magnetic field calculations. So normally the Gauss including atomic orbital is frequently used in 15 nitrogen NMR analysis by computationally. At present, the DFT and also MP2 methods are popular, and the DFT method applied with B3 leaf functional predicts quantitatively the correct values for NMR parameters theoretically. So GeoNMR calculation by using DFT, B3 leaf as a basis set for NMR parameters. So after calculating this NMR spectroscopy, this is how the spectra looks theoretically. So as we can see, the referencing, it has been done through liquid ammonia, so it appears at 0 ppm. This is theoretical NMR spectra by computational GeoNMR calculation. So and it generates that ppm values for different types of nitrogen environments. As you can see, nitrogen 9 it resonates at 356 ppm, and the other nitro group 12 it resonates at 350, and other nitro group 6 it resonates at 353, and the pyrrole nitrogen it resonates at 149 ppm. So these are almost matching to the table what we have summarized for a chemical shift ppm ranges. As we studied pyrrole like nitrogen, it will come around 140 to 160 ppm range. So the pyrrole nitrogen here it is coming at around 150 ppm, and you can see how nicely this theoretical studies will give NMR spectra of 15 nitrogen. So I'll show you some of the experimental results. So I have studied for a few compounds during my PhD work, and these are the results. And as you can see, the beautiful 15 nitrogen spectra and the intensity values are so good, and we can see completely signal to noise ratio by increasing the concentration of the sample here. So for getting this type of spectra, we need a sample of at least, as per my knowledge and my understanding, the instrument what we have 600 megahertz at our department, and I have used for all the compounds at 100 mg scale, and I have dissolved the 100 mg scale in 0.5 ml of the solvent, and then I have recorded these spectra for more than 1 day. So as you can see in this we can get to total scans 5,531 scans. After 5,531 scans, this is the spectra I got for this N-methyl trinitropyrrole, and I have given relaxation delay. Here you can see relaxation delay of 15 seconds. For one scan, it takes 15 seconds. So most important thing for 15 NMR data, sample, a more concentrated sample should be used. So as we have 0.2 mm NMR tubes with us, so in that 0.5 ml solvent I have used, and the maximum amount of compound which can dissolve in that 0.5 ml, that I have taken, and I have recorded this 15 NMR spectra for 5,000 scans. So this is the accumulated scans for 5,000. This is the spectra appears for N-methyl trinitropyrrole. As you can see, I got all the nitrogen peaks, and this spectra I have referenced through nitromethane. So I have converted liquid ammonia spectra into the nitromethane standard reference. So that's why I got all the peaks in the negative region. As I discussed earlier, nitromethane, it may give positive or also negative, but liquid ammonia always gives all positive values only, because it starts at 0 ppm the referencing ammonia peak will appear at 0 and the remaining all other fields will come at downfield. So this is the spectra for N-methyl 2,3,4 trinitropyrrole, and this is another isoisomer of N-methyl 2,3,5 trinitropyrrole. So we can differentiate, and this is also recorded for 5,252 scans, and the relaxation delay is 15 seconds. And this is N-methyl tetranitropyrrole. As you can see, because of same environment of beta nitrose and alpha nitrose, we only get degeneracy 2 for one signal. You can see N1 and N2 and pyrrole nitrogen ring. So here degeneracy is 2. And this is
I think
also
Vikrant,
for,
there is a question from the participants.
yeah, yes, sir.
I can see that.
You just tell me the question, sir.
Yeah, it's as if the compound solubility is very less, then what is the solution?
Compound
So-
solubility is less. You can use the maximum DMSO-D6. So the main solution we can get is quick analysis. We can go for HMBC. So it requires very less sample and you can get the result within 2 hours. But if the compound solubility is very less, the maximum polar solvent what we use is normally DMSO-D6. So with that, how much it dissolves? So you have to increase the number of accumulated scans. So you have to go for 10,000, 15,000 scans. That is the only option what I have observed during this spectral analysis. So main is go for relaxation delay, more time, and then total number of scans should be increased to get the spectra. Or you can go for HMBC analysis. As you can see, if that nitrogen, if it is having only proton interaction, then only it will appear in that HMBC analysis. That is a quick analysis part, you can get it. I think I answered that question.
Yeah, I think it's okay. You can go ahead. If some other question comes, I'll let you know. I'll read out.
Yes. Thank you.
Yeah, you can continue.
Yeah. So I have showed the spectra of N-methylated compounds. Now I am showing the spectra of N-emanated compounds. So that pyrrole nitrogen, it contains another nitrogen, that is through NH2. So as you can see, this pyrrole nitrogen resonates at 164 ppm, and the amine which comes at fields, that is 75 ppm. And the remaining nitro peaks will appear in the range of 350 to 400 ppm range. So aromatic nitro groups will resonate at more than 300 ppm range. And this is also I have recorded for 6,000 scans. Though my sample was soluble in acetonitrile, but it required 6,000 scans to get this beautiful spectra. So and then NHNO2. So that trinitro, it was there, and then NHNO2 type of compound, nitramine derivative of nitro derivative of pyrroles. So in this you can see the NH fifth nitrogen, it resonates as 200, and this again sixth nitro groups, it resonates at 343 ppm. So this is the spectrum for NHNO2, which contains that NH is attached to the nitro group, so it resonates as 196. The nitrogen which appears in the ring, it resonates as 157 ppm. So it is exactly, we can correlate the de-shielding and shielding effects of functional groups which are attached to the nitrogen. So you can think in that way and you can assign. If you face any difficulty in assigning those type of compounds, you can go for computer assisted things by calculating GeoNMR by using DFT and B3LIP method. So there you can get all the peaks with the degeneracy and you can assign the peaks exactly matching to that. So this is salts, ammonium salt of nitro derivative of pyrrole. So as you can see, this ammonium. So I told you liquid ammonia, it resonates at 0 ppm. So this ammonium ion which is attached to the nitrogen, so it resonates at 18 ppm. So the effects will be there on the nitrogen of functional groups. And remaining peaks, because the nitrogen it is having more tendencies, it resonates at 248 ppm. The pyrrole ring nitrogen, and then nitro groups, it resonates at 350 ppm range. So these are the different types of compounds that we have studied for recording this 15 nitrogen NMR, and also it supports our data like proton 13C. They are very important in analyzing our NMR. When you don't have any protons in your compound, so most of the nitrogen nitro groups are there, nitrogen groups are there, then you can go through the analysis of 15 nitrogen NMR spectra. But the main important point is, so signal to noise ratio is very less because of low sensitivity. So you can increase the sensitivity by increasing the concentration of the sample, so as much as available. So as much as it dissolves in 0.5 ml of the solution. So you can use DMSO-D6 as a polar solvent for all the compounds, and you can do until unless if the compound does not decompose in DMSO-D6. If it is more sensitive towards DMSO-D6, and we can, I'll prefer CD3CN. As you can see, estonityl D3 I have used. So estonityl, this is a 239 is a solvent nitrogen. So the cyanide group which is present in the CD3CN, it resonates at 239. So you can see all the peaks completely. So this is a very interesting technique, and you can use it for more nitrogen containing compounds, like you can go for biochemistry also. You can peptides, you can proteins, so where pyrimidines and purines derivatives are there, so you can use it, this type of technique for analyzing those spectra, those type of compounds, and also nitrogen rich compounds. I think I am done with my presentation. If any questions, I am happy to answer.
Yeah, thank you, Vikrant. So the session is open for discussion. Yeah, I think there is one more question from Narendranath Pati. He's asking, is there any option for solid state NMR?
Yeah, if you have the facility of solid state NMR, you can record. So your compound you can get back. If it is solubility is less, you can do it 15 at NMR. But for that also, solid state NMR normally it won't give narrow peaks basically. So solid state NMR also sometimes it gives broader peaks only. So it depends. So the solubility of your compound. So if it is more solubility is good, you can go for a solution state NMR. If not, you can go for solid state. There is nothing problem, and a little broader peaks will appear, as what I have seen during my study.
Yeah, and the machine where this liquid spectra is recorded, there in fact 3 different probes are there: 1.2, 3.2, and as well as 4 mm probes are available with this machine actually.
Yeah.
So any further questions? Students, our students, NIT students? Yeah, Narendranath says thanks.
Thank you.
Any question from NIT students?
If you want to get back your compound, so go for methanol or acetone D6, methanol D4 or acetonityl D3, because it requires a lot of compound. So minimum 60 mg I have recorded, but 60 mg compound it took me almost 1.5 days to get the spectra. Need spectra.
Okay.
That is what you need, more number of scans. That is relaxation delay and more number of relaxation delay and scans. Your spectra will look very beautiful.
Okay. So any more questions?
I once again thank you the organizers for giving me to represent to give about my work carried, especially on 15 at NMR, because I am working on nitro compounds. So
Yeah, so this would be actually very useful. This table, which gives almost all of most of the nitrogen compounds which you would be coming
yeah,
across.
simple
Yeah, this is
alkylamines, arylamines.
from which book? Book or?
Yeah, I forgot to put reference.
Okay.
That reference Just I will add it. Just if anybody wants to note it, you can note that reference. Just
Okay.
1 second, 1 second. I'll add my that reference to this.
Okay.
Is current organic chemistry 2002. So 15 nitrogen NMR spectroscopy in structural analysis.
It's a book or it's a paper? Book only.
It's a no paper journals.
Journal only. Okay. 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 Okay.
So you can note down this, and if you want to read more about this, I did not give any technical details because it covers only instrumentation. So actual experimental lies after the record of the data. So this is current organic chemistry. You can read this one. It gives all the tables, so referencing standards, everything. So I'll show one spectra. This is how after recording through geol, I think NIT Calcutta has geol. So after recording, this is what the spectra it appears. So
This is that ammonium salt, I think.
this is ammonium salt.
Okay.
I can assign the peaks.
Yeah. You can just slightly increase the height intensity. You can just increase. Okay. Yeah, it's
Impurities also it will find if you have any impurities. Any kind of the
here impurity peak is there? No. I think
because we have used 100 mg compound, so you can see no impurity peaks. So compound is completely clear. Thank you all.
Yeah.
Thank you, sir.
No more questions. Then
I think my second session completed here.
Yeah. Okay, that's fine. Okay. You have no more presentation, right? This is
this is final. This is final.
Okay. Yeah, so let's thank Dr. Nagarjuna and as well as Dr. Vikrant for giving an introduction to this NMR analysis, especially the C13, some of the C13 examples case studies, and as well as some of the 15 nitrogen case studies were presented. So if any of the participants have any questions on how actually the experiment is done or the data is collected, so they can in fact contact them. They'll be helping you in identifying that one, so that you can do it. So with this, I thank both the speakers and also to the secretariat. We are coming to the end of today's sessions. Okay, so that also you can announce it. Secretariat, control room.
It was really an interesting lecture. Thank you very much, sir. Dear participants, with this, we come to end of day 6 of the online workshop. For tomorrow's session, please join with us at 10 a.m. Thank you.
Thank you, Vikrant, and thank you, Nagarjuna.
Thank you, sir. Thank you.